Good afternoon, everyone. Before we start this session, I thought I would share some great news, really hot off the press, with the community, and it very much links to the topic and theme of this session. So we found out at midday today that the conditional approval for Translarna was renewed for the next year. So this is really fantastic news. So I just thought I would tell you it's hot news off the press, and it very much relates to the session and some of the speakers which they're going to present today. So that's great news. I would also like to introduce our fantastic panel of speakers. This is the first time they're presenting at the Action Duchenne conference. We're so delighted to have them here. It's really, the session is about access to medicines and how each stakeholder interacts with each other in terms of trying to find potential treatments, but also how the mechanisms of access. So our first speaker in the panel is Nathalie Bere from the European Medicines Agency. She's a communications lead and she's going to present a bit more about the EMA. Thank you very much.
So good afternoon, everyone. Thank you. Thanks for coming to listen. So as mentioned, I'm going to explain a little to you about the European Medicine Agency and also about what is the regulatory approval process for a medicine in Europe. And then the second part is really how patients and carers can then get involved in that whole process. So a lot of people haven't actually heard of the EMA, the European Medicine Agency. We're definitely not as well known as the FDA. And I often say to people when they ask, who are we? I say, well, we're the equivalent of the FDA without the food bit. So basically we're the authority, part of the European Commission that then assesses, authorizes and monitors medicines which are then valid across the whole of Europe. In Europe, there are basically two main ways that you can get an authorization. So when a pharmaceutical company has a new medicine that they wanted to have it approval so they can put it on the market, there are basically two ways that they can do so. And it really depends on what the medicine is. Some medicines legally have to come to the EMA for an approval. So the first way is what we call the centralized procedure, and that is what the EMA does. And this means that when there is an approval, it is valid throughout the whole of the EU member states. The other way is where companies can go to just one or two specific countries and ask for approval there. But as I said, it really depends on what the disease area is. And for rare diseases, they have to come to the EMA for an approval. The basic benefit for the centralized procedure really is that there's one marketing authorization application. So basically the company submits one dossier, there's one evaluation, and if it's a positive approval, then that is valid automatically straight away across all EU member states. So they don't have to go to the other countries to get an approval. And it also means that all the written literature on that medicine, so the package leaflets and the SPC, is the same in every country. It's translated into whatever country, whatever language that country has. So it means that it's harmonized across Europe, which is very important because before the centralized procedure, there were many differences between the different countries on the way that medicines were used. So it's much better to have it harmonized. And you can also harmonize the way that the medicines are monitored as well. So we can look at what are the safety aspects once the medicine's out on the market. This is just to really show you what is the medicine's regulatory life cycle, we call it. So it's a sort of a whole way that the medicine goes into the regulatory perspective. So we have the section which is pre-submission. So this is before a company even submits an application. So they can come to the EMA for certain things. For example, orphan designation. So if they're developing a drug for a rare disease, if they get orphan designation, they then get incentives when they want to submit their application. So they would have fee waivers, they may get scientific advice, other aspects that would obviously incentives for developing this drug for orphan products. Scientific advice, this is something the agency offers on the development plan and the pediatric investigation plan. This is where they have to do investigations in pediatric trials as well. Evaluation, so this is the phase when the EMA is looking at whether or not to approve this particular medicine, and then post-authorization is once the medicine's been authorized and it's on the market, we keep monitoring it to make sure that it remains safe and that it works as we expected it to work. Because obviously when it comes out of a clinical trial, you've only got a certain amount of data. Once it's used afterwards, you get a lot more data from a lot more patients, so you can really monitor to make sure it's working okay. The way the EMA works, so to make sure that we get the voice of all those member states, so you imagine Europe's pretty big, a lot of different countries. So the way it works is that we have a series of committees, and each committee has a defined task. I won't go into them all, but each one has its task, and on each of these committees you have representatives from every EU member state, and each one has an equal vote. So when they're coming up with their opinions, they all vote, and then we make sure that way that it is really the voice from across Europe. Now those committees in turn, they then need help from other external experts. So they may go to the different national agencies, or from learned societies, also from academia, healthcare professionals, and patients and consumers. All these people feed into those committees and give them their advice and their knowledge. And the way it works very simply is that the committees come up with an opinion. So the CHMP, which is a committee that authorizes the medicines, may say, yes, we would give a positive opinion for this approval. This doesn't mean it's valid yet. It has to then go to the European Commission, and they give the final rubber stamp, and then it becomes valid to be used throughout Europe. This was just very quickly, I won't go into it because it's very complicated, but just sometimes people often say to me, why does it take so long for a medicine to be authorized? And this is just to show that there's a whole legal procedural timeline for a marketing authorization. And day one is basically the day the company submits the dossier. We then have different times when we come up with assessment reports. So the committee will come up with an assessment report, and they will be looking at the benefits and risks of the product and the product information, all these different aspects. And at certain timelines, and you'll see here where I put the stop clocks, the little stopwatches, this is when the committee will come up with what they have as a list of questions. So they've got some questions that they want the company to answer. When they send those questions to the companies, the clock stops. So this legal time frame, because we have to authorize, give an opinion by 210 days, but when this clock stop happens, that time, that ticking clock stops. So the company goes away and they need to find the answers to the questions we've given them. Usually they should do it within 3 months. They can ask for an extension another 3 months, which is 6 months. Exceptionally, they may need another 3 months, and you're looking at 9 months. And there are 2 times during the procedure where they can, and it can actually be even more, when it is the times for these clock stops. So that's just explaining why sometimes it is longer than the 210 legal time frame. It really depends on what are the kind of questions that need to be answered and how long it takes the company to come back and satisfactorily answer the questions that the committee has. There are different kinds of approvals as well. We have the standard approval. So this is where the comprehensive normal set of data has to be submitted by the companies. The committee looks at it and then will approve or not. But obviously they realize as well there are times when it's just not possible to get the standard amount of information and data that is needed. So one other way is the exceptional circumstances, and this is when the full set of data cannot be provided. So for example, if they're just it's a very rare disease, there are very few patients, so it's just very difficult to get a certain amount of data. So they can get an approval under exceptional circumstances on a limited amount of data. There's also conditional approval, which you heard was recently for Translana. This is where a full amount of data has not been able to be provided straight away, but we feel with further studies there will be more data coming up. So we give an approval conditional that they will do further studies and bring further data to us, and then this is renewed on an annual basis when we look at the new data. Also as well, there are a couple of incentives that the agency has put in place to really try and accelerate the access to medicines, especially when there is such unmet need. So very quickly, 2 of these, one of them is adaptive pathways, and this is where companies can come for an authorization on a sort of restricted population, so just a small amount of maybe the treatable population, but it means that they could maybe get a quicker initial authorization, and then what happens once it's used on the market, they then do further studies and they can come back and ask for a wider extension. And the other one is PRIME, which is a recent initiative, and this is where if the drug application meets certain requirements, i.e. that it's an unmet need, then they can be given really one-to-one support throughout the whole process. So someone from the EMA is dedicated to this particular pharmaceutical company to really help them through the process to try and make sure it's as quick as possible, meaning a quicker approval if it's positive. What we know at the end of once a product has been approved, it doesn't stop there. Our whole post-authorization scenario is very extensive. So what we know at the end of a clinical trial program is basically the tip of the iceberg to what we will know once the medicine's out there and it's being used by a lot more patients, as I mentioned before. So we have a whole system in place to monitor the medicines to make sure that they are safe and they're working as they should. And we have a dedicated committee, the PRAC, which deals with all of these things. And sometimes they can revoke a medicine authorization, they can suspend one, they can change the indication if they see there are safety concerns that they didn't know about initially. Now to the important part, the patient voice. So how do patients get involved in all of these processes? Well, I'm happy to say that they get involved pretty much all along the life cycles of the medicine, and it has been a progressive journey. We started working with patients way back in '95, but it was just limited dialogue. And as we've gone along, as the years have gone by, we've set up a formal framework of interaction. We have a dedicated working party that meets 4 times a year. We've really tried to learn by experience and make sure that we can get that patient voice into the different perspectives along the way. But this highlights really just that we've progressively tried to improve and expand as things go along. And this framework I mentioned really has 5 elements. The only way that we can work with patients and carers is by having these 5 elements. So we need to have a network of patient organizations. We need to have a forum where we can exchange information with them, so our working party. But we also need to have a pool of individual patient experts who are willing to come in on an individual basis and contribute to all those ongoing scientific discussions that are happening and to share their experience and give us their opinions and their knowledge from living with the disease. We also need to interact into the whole EU regulatory network, so within all the different agencies in the different EU member states. And also, we need to make sure that every patient comes in, that they are properly prepared and trained and ready to participate. We can't expect you to come in and just know what to do and what is going on. So we offer a whole system of training and support. We have webinars, we do training days, and one-to-one support for the patients coming in. So it's very important as well to make sure that you're properly prepared. Any patient organization can register with us. There is certain criteria they need to meet. These are on our website. And also any individual patient can register on to be in our individual database. So there are 2 things. There are eligible patient and consumer organizations and a database of individual patients who are interested to be involved with us and interested to receive information on a specific disease area. This just shows you some of the organizations that are already on our list. Now, some of these are disease-specific, some are have individuals as members, others have other organizations as members, but it's very, very flexible. Now, this slide really was just to show you, again, you can see this regulatory pathway, so the pre-submission, evaluation, and post-authorization, and the little orange blobs really just show you where we've put in place opportunities for the patient voice. So along the bottom you can see this is the different committees, sorry for all the acronyms, but these are basically all the committees along the bottom, and this is where we have the involvement of patients, so along the life cycle. And patients themselves come in, or carers, and they work in different ways. So sometimes, depending on the activity, they may come in representing all patients. Basically, it's just a patient representative. They may be coming in to represent their particular organization, or they may, in other cases, coming to represent themselves, their own experience and knowledge. It really depends on what the activity is and also the level of confidentiality that is associated with that. So currently we have patients who are members of our management board and members of 4 of the committees. When I say members, they are full right voting members as any other member. This is the Patients and Consumers Working Party I mentioned earlier on. So we have 20 patient organizations who are part of that. We meet 4 times a year, and they're our sort of first port of call when we are looking for patients to participate in other activities. They're also invited to all of our conferences and workshops. They respond to consultations we have on our website, on guidelines, and any other EU-wide initiatives. Really, we try and involve them everywhere. And also mainly, the most important thing I think is on the actual medicines evaluation. So in the development phase, so this is these scientific advice procedures. So the companies come and asked us for advice on their development plan, the design of their clinical trial. We will have a meeting with the company and we try and invite a patient as well. So a patient could come in and give their perspective. Do I want to attend clinic 3 times a week? Is it feasible, the kind of things that they're proposing? This is where we need your input to make sure that it's realistic what is being proposed in terms of the clinical trial. And then once we have the evaluation going on, we have different scenarios where we might have an expert meeting. The committee's decided I need some extra information. We do a meeting with specialists from across Europe. Again, we bring patients in on board as well. Sometimes there might not be enough time, so we might have to do a consultation in writing. We get a list of questions and then we send them to you in writing and you respond that way. Also, all of the written communication coming out of EMA is also reviewed by patients to make sure that it's patient-friendly, to make sure we're not going too much into the technical jargon, but that it is really understandable for the lay audience as well. I'm not going too long. Just to show you, I mentioned the progressive journey, so this just shows you the numbers of patients that have been involved across the year. So last year we had over 700 patients involved in the different activities across the agency. We also try to make sure, well, what is the impact of having patients? People often say to me, so how do you know it's of value? How is it beneficial? Now, we do questionnaires. We send questionnaires after the activities, both to the patients themselves and to the regulators to make sure that it is working in the right way and everybody's happy with the input. And for the scientific advice, we did some evaluation as well up to 2014 on when a patient came to one of the scientific advice meetings and they said something, how often did something they say actually make a difference to the final report that was sent to the company? And you can see that it's almost 50% of the cases when something was said. And now this doesn't also take into account when patients actually agree with what's on the table. That is also a positive contribution. And the other one as well on the written communication. The European Public Assessment Report, which is a report the agency publishes at the end of an authorization, explaining how it was authorized. And patients reviewed these, and 70% of their comments were actually taken into account and changed in the final written document. Obviously, there's been challenges along the way, I think, from both sides. It's not always easy to be able to find patients and carers who have time to come in and participate, who are willing to give their time to learn what's going on, to read all the documents that are involved in whatever procedure that is. Coming from across Europe as well, sometimes there are language barriers. We need to make sure, as I mentioned earlier, about this proper training and support that the patients are given. Also, it's very important that there is a defined role. It's very important that patients and the others around any given table, any given meeting, understand what is the role of a patient. You're not expected to be following the scientific elements. You're there to give your vital, crucial knowledge as a patient or a carer living with a disease on a daily basis. That's what we don't have on the table. That's what's missing from the data that we already have. So that's what you bring in. Sometimes there's the issue of conflict of interest. Obviously, we need to make sure that they are not on the one hand discussing the product with the pharmaceutical company and then coming into a meeting with us. So every person who comes to any meeting has to fill in a declaration of interest and a confidentiality agreement. And representation as well, just making sure that this clear role when you're coming to a meeting, what you're representing. And just finish up by saying the value that patients bring into the discussions. I think I've mentioned it earlier, but bringing the everyday aspects of living with a disease, telling us what is important to you. I mean, if there is a very slim amount of benefit for a given drug, on paper, this may look insignificant, but when you come in and you explain to us, well, that very small amount on paper is a huge amount to me or my child on a daily basis. That increase in movement or whatever it is very, very important. And that might not show on paper, but it shows on your daily interactions and daily life. And that's what you bring to the table, and that's what you explain to the regulators and help them make the best decision for everyone concerned. And that's why here we say means more meaningful regulatory outcomes for all concerned. And hopefully it also improves the information and communication to patients. And today, patients really are a recognized and integral part of the agency's work. They're systematically involved across the board. That doesn't mean that it's perfect. There's always work to be done, and we always want to hear from patients how we can do better, where should we improve, to make sure that we can, do it and everybody's happy from both sides. I think that's it. Patients complete the jigsaw. Thank you very much.
Thank you very much, Natalie. It was a great presentation, a good overview. I would like to introduce the next speaker, who's Dr. Daniel O'Connor, who is from the MHRA, and he's going to talk a bit about access to medicines and how he's working with the EMA.
Thanks very much, and it's a real privilege to be here. And I know it's always difficult post-lunch, but I know that you're getting coffee afterwards, so hopefully that keeps some interest. So today I'm going to talk about a UK-specific scheme and try and put it into the context that we've nicely heard about the European Medicines Agency. Just a show of hands, how many people have heard of the UK Early Access to Medicine Scheme? Oh, great. So that's a really good number. So that makes my job a bit easier. I've just got a few introductory slides really just to set the scene. As Natalie says, I work at the UK MHRA, and we work in this regulatory network. So we work within the European Medicines Network framework, and we're based in London, we're based in Victoria, and we aim to protect and improve public health, and we're one of the largest regulators in Europe, and we aim to be an accessible, pragmatic and efficient medicines regulator. So in terms of early access, I know this will be at the forefront of your mind in terms of making sure that medicines, when they're developed, are able to be there for your children. A key challenge, I think, is facing us in terms of the balance between approving medicines when it's perhaps a bit too early, but when actually we don't still have quite enough data. But as Natalie said, there's a number of ways that as regulators we can sort of leave that uncertainty. So we've got the uncertainty, but we have regulatory pathways to try and help with that. But there's always going to be this so-called evidence versus access balance. And really excitingly, I think, for regulators, there were three new initiatives that came through in the last 2 to 3 years. And you've already heard about these 2 European initiatives, the Adaptive Pathways and Priority Medicines Prime. In the UK, what we've initiated is called the UK Early Access to Medicine Scheme, which aims to give patients with life-threatening condition access to medicines before the medicine is formally licensed. So I'm just going to now cover some of the history of where the scheme came out from. And these are some of the really important milestones. Back as early as 2008, there was a recognition from the Ministerial Industry Strategy Group that an early access to medicine scheme would be important. This was followed up later through the Prime Minister's Strategy for Life Sciences in 2011, where there were 2 components to that were related to the early access to medicine scheme. One was an EAMS public consultation, and one was the formation of an expert group on the innovation in the regulation of healthcare. Quite a mouthful, but quite an important group that sat for a year. And I'll cover both of these briefly in a couple more slides. And really excitingly was when the MHRA launched a scheme in 2014, in April 2014, and we have a 2-step scheme, a Promising Innovative Medicines designation and an EAMS scientific opinion. And I'm going to cover these in a bit more detail in the following slides. And as milestone points, our first PIM designation was given in September of 2014 with our early access to medicine scientific opinion, which relates to patient access in the NHS before products licensed in March of 2015. So the Ministerial Industry Strategy Group looked at the question of early access to medicine, and their regulatory working group considered the options. In conclusion, what they felt was that whilst acknowledging for most medicines that it would be towards the end of the development program, there could still be potentially life-saving treatments that could be given to patients up to a year earlier. In terms of the public consultation that was launched in the summer of 2012, it ran for about 4 or 5 months, and there were 26 questions. Importantly, the question about whether it should be put forward or not was one of the questions, and there was overwhelming support for the scheme. And I've just really summarized at the bottom of that bullet point really what the conclusion from the consultation was, that EAMS addresses a public health need to improve patient access to important innovative medicines for patients with life-threatening or seriously debilitating conditions without adequate treatment options. And if you want to read more about the consultation report, I've provided the link. As I said, this expert group was formed from a number of stakeholders, but importantly, the group also included representatives from organisations such as Cancer Research UK and some patient groups as well. And the group, one of their tasks of the group was to review the early access to medicine scheme proposed response to the consultation. And there was overwhelming support for the response to the public consultation on EAMS. And again, there's a link to that document. So what happened at launch? So this web page here, you can see this is our current web page on the MHRA's home site that you can go to, or it's very easy to Google to, but the link is there. And we have a supportive framework in terms of guidance documents and templates to support applications for the Early Access to Medicine scheme. But in an overview, as I've said, it's a two-step scheme, the Promising Innovative Medicines designation and the EAMS scientific opinion. Its aim is to cover medicines that don't yet have a marketing authorization. So as Nathalie explained, the medicines that go through for rare disease go through a marketing and licensing process through the European Medicines Agency. But when there's a gap between access or perhaps clinical trials are finished, this is where the Early Access to Medicine scheme could perhaps still provide valuable access to patients for these medicines. So it's about unlicensed medicines. It's not a substitute for appropriate clinical development, and it is primarily aimed at medicines towards the end of their development. Though I would say what's really important is to be able to demonstrate a positive benefit risk profile. And during the scheme, the company provides the medicine for free of charge. There were 4 EAMS criteria. These are listed here, but essentially has to be a life-threatening or seriously debilitating condition, that the medicine offers promise over and above existing therapies, that the benefits are likely to outweigh the risks, and the company can provide the medicine according to a high quality standard. In terms of a bit more on the Promising Innovative Medicines designation, it's issued by the MHRA after a scientific advice meeting. And what the PIM designation gives, and hopefully companies and academic units come much earlier in their development program, it allows the company to get some reassurance and dialogue from the medicines regulator that their clinical development program is on track, so they have an early review of their data. But importantly, for those products that are given a PIM designation, they'll also get specific contacts in the NHS and HTA bodies to start engaging much earlier about patient access issues. The EAMS scientific opinion, which is the second step, which is given after a timetable of either 75 days or 90 days, which is very fast, will equal patient access in the NHS. And what the scientific opinion does is it describes both the benefits and the risks of using the medicine, which supports both the prescriber and the patient in making a decision to use the medicine before it's formally approved. There's also additional interaction not only with the MHRA but the HTA bodies and NICE, for example, where PIM designated products are prioritized through NICE selection topics, and also EAMS products are planned as priority work in the work program. So what's happened since launch? So as I said, the scheme's been going now for about 2.5 years, and we issued our first scientific opinion, which was for melanoma, and all of our scientific opinions are published in this format, where we have a brief public assessment report, which is written in lay language. We have a treatment protocol for patients, which is written in a language that patients should understand about some of the risks that might be involved with the medicine, a treatment protocol for healthcare professionals, and then a treatment protocol regarding pharmacovigilance, so extra data collection for safety to ensure the medicine remains safe during the EAMS scientific opinion period. This table just illustrates the numbers of applications to date, and recently this has been updated, and we've now had 36 applications for PIM designation, and we've issued 23 PIM designations since we've launched. In terms of the EAMS scientific opinions, we've now given 10 EAMS scientific opinions. The majority of these have been in the field of oncology. In terms of the learning process that we're still doing, what we wrote is with the first company that went through the EAMS scientific opinion, we wrote a case study, and this is freely available in this link here if you want more information about what actually happens during an EAMS scientific opinion. But again, one of the conclusions from the case study is that EAMS is an important step in ensuring that patients gain access to innovative medicines as soon as possible, improving health outcomes in patients that early need urgently new treatments. And in this case study, the company provided data to suggest that they had over 500 patients benefited before the product was actually licensed, and that's obviously a really fantastic event. There's a number of things that are ongoing now in terms of evolving EAMS. Now we've learned some aspects over the last 2 or 3 years, two years. There's a government EAMS task force which was launched after we went live about in the summer. And this group is a number of industry representatives. There's also representatives from some of the trade associations. And within this group, what we're looking at is ways that we can improve the process of EAMS. In addition, there was an independent review of EAMS by PricewaterhouseCoopers, which was commissioned by the government, and they published a report back in March. And in this document are a number of recommendations that are divided into short-term, medium-term, and long-term. And there are a number of things that these will take into consideration in the following report. This UK accelerated access review, which aims to speed up access to innovative drugs, devices and diagnostics for NHS patients. And what this review will do will take into consideration, or has taken into consideration, that independent report, and it was published very recently on the 24th of October. EAMS, the Early Access to Medicine Scheme, is included as a specific term of reference, and a number of recommendations have been made in that report, including that patient access could be brought forward in the UK up to 4 years if an EAMS scientific opinion is used, saving from the EAMS component 12 to 18 months. In addition, as I mentioned earlier, EAMS medicines are provided currently for free of charge. And one of the recommendations was that for SMEs and not-for-profit organisations with products on the pathway, there should be in some cases some form of funding. And patient involvement in the accelerated access review is a really key component. So to summarise, EAMS gives patients early access to medicines that do not yet have a marketing authorisation, so a drug licence, when there's an unmet medical need. Patients are able to benefit from important medicines before they're formally licensed, and the prescriber and the patient has greater confidence in using that particular medicine. In developing the scheme, users of medicines have been consulted at different stages, including at public consultation and in the expert group. And our assessment process, the assessment process in the MHRA, involves consulting our expert committees, which include practicing NHS clinicians and input from lay members. And as I've mentioned in the previous few slides, EAMS continues to evolve with a number of initiatives. Thank you very much for your time.
Thank you very much, Daniel, for a great presentation. So our next two speakers, Sheela from NICE and Edmund from NHS England, are going to give verbal updates about some consultations and some updates from their side, and then we'll have the final speaker updating us on the ABPI. Thank you.
Diane, thanks for inviting me to come and speak today. I thought it might be helpful to start off with a little bit of a description of what NICE does for those of you who've not heard of us or don't know what we do. But in essence, we follow the regulators once they have had an opportunity to look at licensing products, and we are then given the responsibility of looking at drugs to see whether or not the benefits that have been demonstrated in trials actually are there, and whether the price and the cost is demonstrated, so therefore being able to define if it's value for the NHS to buy those drugs and make them available for the NHS and for patients who access them. So that's the kind of role that we play. Some of you obviously know that recently we evaluated Translarna, or Atalurin as it's normally known in some areas, and so that's one of the sorts of roles we play. So I wanted to just have a really quick summary and run through what my role is at NICE. So predominantly my role is to run a program that looks at drugs for rare diseases. That role is quite a challenging one, and we have a committee that looks at that, and the committee is made up of health economics people, clinicians who operate in the rare disease space, and quite unusually patients and lay members who actually have rare diseases giving some opinions around the drugs that we evaluate. We also have the opportunity for patient groups and patients to participate in the meetings when we're evaluating those drugs, and again it's quite a unique scenario for us to be in. The programme does look at very rare conditions, as I said, and we would be looking at trying to capture drugs that work for patients where the treatment is normally concentrated in very few centres. So Edmund will talk to you a little bit about the highly specialised services within the NHS, but normally patients with these rare conditions are treated in very few centres who have a range of expertise to offer. The condition that we would normally look at would be very chronic and disabling, as in this scenario. The technology would be seen to be operating in a context of highly specialised service, and generally these drugs are very high cost for the NHS. And it's a lifelong treatment for a lot of patients. So those are the sorts of criteria that we use to identify whether a drug should be looked at by the highly specialised technology programme. With the rare disease scenario, it's really challenging for us to undertake these evaluations because a lot of the time there are a very small number of patients that are affected by it. So there is not a lot of information out there, or what's known as natural history, to enable to see whether or not these things that the company who are presenting the drugs to us saying actually are happening. A lot of the trials that companies undertake are for a very short duration, so we're unable to see the 5-year, 10-year effect. They're wanting to try and get the drugs out there quickly. They've got a small population, so we've not got a lot of data to look at. They are very challenged in trying to get a lot of consensus on what's out there as the best treatment currently available from clinicians as well. The other things that the committee also tries to consider when they're making evaluations is the societal value. So what does society value if this drug was given to these patients? The seriousness of the condition and the availability of other treatment options. What is there already being offered to these patients, and is what is being proposed better? And then things around the carers and how it impacts on families and carers are also taken into consideration. Now, all of this is a really difficult discussion that happens at committee, and that's the reason we've got a committee that's got quite a lot of range of expertise. We often find it really hard to define the patient population and be clear about how that will work. And we often are faced with what's known as uncertainties in the evidence because of the trials being conducted in that small population and a lot of assumptions being made as to what the long-term benefits of a drug might be in these rare conditions. And it's also very difficult sometimes for the committee, the highly specialised committee, to reconcile the uncertainties that are demonstrated in the drug against the value of the drug and the cost to the NHS. And the reason that becomes an issue is because of the high cost that these drugs are for patients and the understanding that the impact that that might have on the NHS budget. Now, I'm sure none of you are unaware of the challenges that the NHS currently is facing in terms of the budget constraints. And I'm not saying that that's it's just a scenario that we are currently in. So the committee for the highly specialised team are very much challenged in trying to ensure that drugs that are evaluated will have a good effect and will be beneficial to patients, but also help the NHS manage that budget. And the reason that I'm raising that now is that recently NICE and NHS England in collaboration have just launched a consultation. And the element of that consultation for the highly specialised arena or the rare disease community is that they have considered thinking about how NICE and NHS England can work together to try and manage that so that they can make access available for patients with rare conditions, and they can ensure that drugs get through the pipeline. So they've proposed some changes to the, sort of the terms and conditions of how these appraisals are undertaken by NICE. One of the proposals around this is that if a drug is presented to NICE that has what's known as the budget impact, so if the cost of the drug and the impact on the NHS budget in the first 3 years is going to exceed £20 million, then the company who is producing the drug will be asked by NICE to go away and have a discussion with NHS England. The reason they'll be asked to do that is to come up with a solution as to how this drug could potentially be made available in the NHS without displacing other commitments that the NHS has already made to other things that they're having to do. It could be a range of options. The company could potentially offer to reduce the price of the drug and reduce that budget impact. The company could maybe suggest identifying how to classify patients with the greatest need and then stagger the access to the drug. The company and the NHS might agree another solution of maybe different commercial terms or different arrangements around access. And we're hoping that if that happens, then actually we're going to be in a stronger position to be able to offer some greater opportunities for drugs to be accessed by patients with rare conditions. Another thing that they are proposing is, up till now, the highly specialised appraisal committee hasn't had what's known as a quality threshold or a benefit threshold that's been applied, that is generally applied to technologies or drugs that are kind of for the more common conditions. And they're proposing that that is introduced as well. So the idea is if the budget impact is within the 20 million and you are meeting this proposed level or threshold and the clinical effectiveness of the drug is demonstrated, then you are automatically given a positive recommendation by NICE, and therefore the NHS would have to make that drug available within the appropriate timescales. However, if any of those things are missing from that puzzle, then there is opportunities for the company and the NHS, guided by NICE, to be able to have some discussions about how that would work. Now, with the Translana discussion, some of you may or may not be aware, but we were really fortunate in NICE and the NHS and PTC to be able to come to some really interesting arrangements of how to gain access to the drug. And we've put together a managed access agreement in that scenario, where in the course of the 5 years of this agreement, the company, the NHS and NICE will be collecting data to produce the benefits and to get some convincing evidence to support the case for continuing to have access to Translana. And the reason for doing that is that the committee, when they were looking at it, saw the promise and they were really convinced by the promise. So they tried really hard to come to a conclusion to try and make this drug available. And the idea around this new proposal is to try and think of innovative ways to ensure drug is given to patients where it's appropriate to do so. And so the reason I wanted to bring that up is I was speaking to Diane about making sure that you've all had an opportunity to look at that consultation and maybe as an organisation respond to it, because I think it's really important. We've clearly got a lot of new drugs coming through into the Duchenne space, and it would be unfortunate if you didn't have that opportunity to participate in that consultation. The other thing that I wanted to just talk to you about is the uniqueness of the highly specialised appraisal process, in the fact that it does actively include patients. So as I said, we do include patients along in the committee. So we've got a lay member. We've also got a member who is a patient representative from a rare disease condition, and we've also got an ethicist on that. We are also looking to, in the future, potentially recruit new patients onto that committee. But the other thing that we also do is, throughout the process of when drugs and topics come to NICE to look at in the rare disease space, is actively seek involvement from patient groups. And Diane hopefully is testament to that in some of the discussions we've had for some of the drugs that we've been doing. So we we do actively involve patients to ensure that we get that opinion and the input in terms of the gaps in our knowledge. And Nathalie described it really well around, giving that kind of context of how those conditions, how they are for you, how they work for you, what is the drug going to offer you. It's helping us fill those gaps. We also are really fortunate in being able to use patient groups to help us understand the patient population better and the impact of the disease better. So we can get the clinical data, we can get all of this, trial data, we can often get lots of information thrown at us in lots of papers and things, but actually hearing it from the patient group and the patients who are experiencing these conditions can be absolutely tremendous for us, and it does play a huge part in the discussions that we have when we have committee meetings. We really value that input, and I'm hoping that we will continue to be able to engage in that manner, because that really does play a big part in the decision-making process. And finally, just to say that you shouldn't underestimate the amount of information you can give appraisal processes and sort of the regulators, as well as what's known as HTA or Health Technology Appraisal Organisations, because we do get a lot of data thrown at us, but actually when patients come to us, they bring those conditions to life, and they really, really do give us a whole plethora of information that we wouldn't get otherwise. But I'm going to leave it there for now and open it up for...
Thank you very much. We will be sending some details to everyone about the consultation, which is very important. Our next speaker is Edmund Jessop, who is from NHS England, and he's also going to link how he works with NICE and also present some more information about that, the stakeholder. Thank you.
Thank you. So as you heard, I'm Edmund Jessop. I work for NHS England. NHS England was formed in April 2013 to take over from the Department of Health, the government ministry, the job of day-to-day running of the NHS. So the situation is that politicians, government gives a strategic direction to the NHS, but the day-to-day running of comprehensive health services for all 55 million people who live in England is the task of NHS England. So that will tell you that NHS England is a very large organisation. We're an administrative headquarters, but the overall task includes making sure that primary care and GPs are working properly, that local hospitals are working properly. And then in respect of specialised healthcare at specialist hospitals, we don't just have a general oversight, but we directly fund the specialist services. So for a patient with muscular dystrophy, local care by the GP and the local hospitals, and the drugs that are given locally in this session on access to medicines, that would fall to the local organisations. And if you've spent your life living in England, it's really difficult to keep up because they keep changing their names. And at the moment, the name for the local budget holders is clinical commissioning groups. But the budget for the whole of specialised care is held by NHS England, and therefore specialist disease-modifying drugs for rare diseases the decision falls to NHS England. In respect of the very expensive diseases for very rare conditions, which would be the case for mutation-specific diseases in muscular dystrophy, we look to NICE for advice. And in fact, there's a piece of legislation, there's a law that says that if NICE says yes, we are then legally required to fund that drug. The consultation that Sheela mentioned that has just been issued has been issued because that system whereby one organisation could tell another organisation how to spend its money, that's worked fine for the last 10 or 15 years, but we're in austerity. The financial situation is much more difficult than it was, and the view is now there needs to be some communication at the very least between those two organisations. the sums of money do seem absolutely enormous. The budget for specialised healthcare that we hold, which covers not just drugs but everything else, is more than £15 billion a year. So 15 with 9 zeros after it. That's the total budget we're talking about. But even so, the requirement to spend an extra 20 million is seen as significantly challenging. I'm a doctor, I'm on the medical side of NHS England, don't deal directly with the budgets or manage them, but that is the situation, that's the scenario we're living with. NICE has this power to compel us legally to fund a particular drug, but of course that is done then at the expense of other things that we might need to spend our money on. We've moved into a situation where we try to make all our spending decisions once a year. So rather than spending money as and when people ask us for new things, we collect up all the calls on our budget, and once a year there's a decision meeting. And just for example, the recent decision meeting had to balance whether we were going to spend money on severely disabling disorders for very rare disease, or cancer drugs, or treatments for blinding disorders, or preventive treatments for AIDS. I think that one, as you may know, got into the newspapers because NHS England made a decision, and then that was challenged in the courts, and the courts said, well, no, that needs to be one of our spending decisions. So it's quite a complicated process. I mention this because this also explains why it takes time. So a drug can get its marketing authorization, can launch commercially, but in terms of the spending decision, whether we can fund the drug, that has to wait till the next annual budget round. The good news, as you know, for Translana is that in collaboration with NICE, we've constructed this managed access agreement, which will fund Translana for every eligible patient in England. We've funded to the license, so the regulators tell us whether a drug is safe and effective. And as you know, the license for Translana is for patients aged 5 and over who are still ambulant. So any person in that category will have Atala and Translana funded by us within the terms of this managed access agreement. All the other speakers have emphasized how intimately patients or their representatives are part of the decision-making process, and that is also true for NHS England. So there are patients or lay people on every decision-making, every advisory committee in NHS England. The design of the scheme that's enabled us to fund Atala was very strongly driven by the advice given at the hearing at NICE, and it's very much driven by the importance of maintaining ambulation in this disease. We have other managed access agreements for other extremely expensive medicines which have different clinical parameters built into them, and they're also, the parameters built into the access agreement are based on advice from patient groups. At the moment, in fact, most of the advice we're receiving is from parents, because the drugs and diseases we've dealt with so far are too disabling for patients themselves to present their own view. Actually, with one exception, I think about it, there was one where we had patients themselves. So again, it's just to emphasize that we do heed the views of patients in all this work. I'll leave it there for now, but I'm very happy to take questions when the panel discussion starts.
Thank you very much, Edmund. And I'd like to introduce our next speaker, who's going to talk about the ABPI, Paul Schofield from Nat Pharmaceuticals.
Thank you. So it's my pleasure to sort of conclude the discussion so far on trying to fit into this jigsaw, what is the ABPI? So simply put, the ABPI is the trade association of the UK pharmaceutical industries. So really it's a representative body. So when the likes of the MHRA, government, NICE, etc. ask for an opinion about policy, about recommendations from the pharmaceutical industry, the voice of the pharmaceutical industry comes from the ABPI, based again in Victoria, in London. The ABPI then is comprised of probably over 100 from small, medium and large pharmaceutical companies, but doesn't specifically lobby on behalf of one particular drug. When it comes to an individual medicine, then obviously that goes down to an individual company. So be it, for example, with Ataloran, that would go to PTC, the company that actually manufactures. It wouldn't be the ABPI that would be actually involved directly to discuss access, to discuss recommendations and submit to the EMA for an approval. That would come to the individual company. So the ABPI really is that trade association, say, that is consulted and feeds into many, many documents, many positions, whether it be access to medicine scheme, accelerated access. That's where on a daily basis the ABPI works. And again, of course, to get that opinion, I'm a medical director of a company based in Cambridge. The ABPI will consult and will send around sort of on a regular basis emails saying, this has come up for consultation, what do you think, what's your opinion? We're forming a paper, a document to send back as part of our consultation ongoing to say what is the position of the UK pharma industry in this particular condition or this particular disease area or approach to licensing new medicines. So what about patients in all this, the ABPI? Well, the ABPI has many groups, many committees like other large organizations, and I sit on a steering committee of the Patient Organizations Forum, which literally is a consultation group between the pharma industry, patients and the charity groups to talk about things which are of mutual interest. But again, not speaking about individual medicines, but highlighting areas for input brought mutually. So the steering committee is comprised of both representatives from the pharma companies, but also from patient organizations. So we look at agendas, we meet on a quarterly basis, we look at interesting topics obviously of relevance to patients, to patient organizations and to the industry, and work together and discuss together and debate those issues. And that's something that is really there as part of the link with patient organizations through the ABPI. It aims to be transparent, and on their website there are the minutes, the meetings, the publications of what's happened and what's been presented at those meetings so that you can all access and read what's happening. And the companies involved in this forum, not every company is involved, but this is just a selection of large, medium and smaller sized companies, including my own, that are involved in this grouping. And finally, really just to conclude, ultimately all of this comes down to discussions around money, around the willingness to pay, and all the aspects of what you're hearing from the getting a marketing authorization, conducting clinical trials, then getting through the health technology appraisal process, and then getting ultimately a medicine to the market, all comes down to what is the cost, what is the benefit, what is the safety, and what is the efficacy of that medicine. So it's around to the payment based on what will the actual benefit be to the patients and can it be afforded within the grand scheme of a limited purse. So I think that's all I really want to say about what the ABPI does as part of this whole grouping. Thank you very much.
And thank you very much, Paul, for that overview of the ABPI. So we have 15 minutes for questions. We really want this to be an interactive panel discussion. This is a really fantastic panel, so do take the opportunity to ask questions. So the floor is open.
Hi. Oh, it's working. There's been a lot about sort of taking into account what patients say and getting feedback from parents and all that sort of thing. And I don't really know who to address this question to, but I'm concerned about children. I'd like to know what steps are taken to look after the interests of children, particularly when they go in for patient trials. I know of 2 trials in the last 4 years that have had to be aborted for some reason. And I've had some very upset parents who've said to me the drug is working for their child. How do you explain to a child that they're going to go in for a patient trial and then have the trial aborted? These are not adults. Who looks after them? And who on these committees take into account their interests?
So thanks for the question. I mean, I think it's difficult to be precise because I don't know the grounds for why the trial was stopped. But I mean, I would say that the clinical trial application, so the process in ensuring that the clinical trial goes forward, is a very robust process in terms of looking at the safety aspects, but also there's an ethical review. I don't know whether in the specific circumstances you're talking about whether the trial was stopped due to a safety reason or not, but I would hope that within the trial protocol and the investigators that run the study, they would be able to communicate to the children and the parents the reason why the study was stopped. So I'm sorry, I can't give you more of an answer than that.
All I do know is that the ones I was speaking to, the trial and the drug was working and there wasn't a danger. From the speakers at this conference, when they came 2 years ago and 4 years ago, it was they hadn't achieved a percentage.
I mean, maybe sometimes it's difficult to recruit studies overall. I mean, there's a number of reasons why, unfortunately, trials don't go forward. I mean, that should be communicated to the trial patients and the trial patients. I agree that's something that's very important. I don't know why that wasn't done in these 2 particular cases, but I hope that would be normal practice for anyone involved in a clinical study, that they would be informed why that clinical study was stopped early.
The most interesting slide, excuse me, the most interesting slide which we just saw was the one with all the pound signs in different colors. And it basically is money that we're talking about, as I see it. Are you taking into consideration what happens if one of those drugs is not given? That we end up with having to look after a child going into a young man with all these difficulties and requirements. I'd have thought that's far more expensive than giving the drug.
I don't disagree with what you've said, but I think as an organisation like NICE, who have to evaluate the drug to check whether it's got the clinical effectiveness that's been described against the cost and against a challenged budget. This group is one group of patients that we have to look at, but we have a duty to look at the entire population and how that balances out. And I think in terms of the managed access agreements that we've tried to put together for Translana, which is just one of a couple, and we're actually doing it for cancer as well, is that we're trying to think about how we can collect information and data to support the decisions around funding these drugs. But as an organisation, we're a public body, so we are answerable to the public, and therefore we've got to make decisions that allow us to help the NHS manage that budget better.
So I agree with all that, but in fact the specific scenario that you gave us, where it is cheaper to give the drug than to provide the care, that we would fund, because the part of the process at NICE is exactly to take all those sorts of things into account. So the difficulty with muscular dystrophy is that we don't have any hugely effective, well, we don't have any cures, that's for sure. But if we were to find a cure, if for example gene therapy were to prove successful as a cure, which it has been in one or two, not many, one or two other diseases, then we absolutely would look at the cost of the drug, or we would look at the price that the manufacturer wishes to charge for it, and we would look at the cost of lifetime care for the disease, and those two would be absolutely part of the way the decision was made.
Thank you.
Sort of a vaguely similar question, really. First of all, just thank everyone for coming along, and having come to these conferences over a number of years, my understanding of the regulatory and funding process has really come a long way. I now absolutely recognise that the regulatory authorities are on our side as parents, and that the regulators are desperate to approve a drug and desperate to try and help families and children like my son. My slight concern listening today is there does seem to be considerable duplication of what NICE is saying in addition to the regulator. NICE talks about effectiveness and clinical effectiveness and all the rest of it. And I always understood that that was the regulator's part. And my understanding from what I've heard today is the only difference between NICE and the regulator is NICE is looking at money. And whilst 3 or 4 years ago I might have thought that the major barrier to my son getting medication was the regulator, I don't think that's the case anymore. And I'd like to just hear a little bit or be corrected that NICE doesn't duplicate the work of the regulator. And I'm just anxious that the regulators are on board, but perhaps NICE and certainly NHS England are putting more and more barriers in between the innovations that the pharmaceutical industry are putting up and all our children getting medication.
So the regulator looks at safety, quality and efficacy, and the difficulty at the end for NICE and for NHS England is efficacy, because we need to know not just that the drug is working, but how great the benefit is. So if there's a huge benefit, that's an easy decision. If the benefit is small in relation to the cost of the drug, then that's when we start thinking about all the other calls on our budget. And in NHS England, we're very grateful for the NICE process, because the NICE process brings out, with huge thoroughness and scientific rigour, the best possible estimate of the lifetime benefit of a product, and in relation to the price that's been quoted. And that is not part of the regulatory process. So that's the difference between the two, and that's why you're right, there appears to be an element of duplication.
So the duplication is all there's a huge amount of duplication. The only difference is the finance.
I'm just going to pass to my colleague from EMA who wishes to comment.
Just a brief add-on as well is that to try and sort of make sure that everyone's on the same board is that we have also the opportunity for scientific advice with HTA bodies. So sometimes when the pharmaceutical companies ask us for scientific advice on their development plan, we can also make sure that HTA bodies are present at those initial meetings so that at least everyone's there at the very beginning. I mean, I think now it's much more about early dialogue with everyone around the table. So already at that initial discussion, when we're talking about the clinical trial, what the endpoints are going to be, what data's got to be looked for, the HTA bodies can also say, okay, this is what we would expect, you know, but this benefit versus this risk, or this is the data that we would be looking for. So having everyone around the table early on also makes sure that everyone knows what is going to be needed later on as much as possible along the approval process.
Yeah, can I ask? Okay. My first question was for Nathalie, just whether you could clarify something. You talked about the prime mechanism EMA and addressing an urgent unmet medical need. Given that Translarna's conditional approval has been extended, does the EMA still consider that Duchenne has an urgent unmet medical need? That's my first question. My second question is to the whole panel. It's something probably more of an lament actually in this awful post-Brexit world that we're facing. And what the panel think about the impact will be. I mean, for us who fund research in Europe, the costs have skyrocketed. That's one short-term disaster. But, you know, when you look at projects like vamorolone that have benefited hugely from EU collaboration, from funding, from the sharing of patient groups, the sharing of expertise and knowledge, how can the panel reassure us that as patients, our children are not going to suffer as a result of Brexit?
So to answer your first question, I mean, we are very aware that there is still a very much unmet need. I mean, Translarna just addresses a very small amount of boys and those that are ambulant and over a certain age. So obviously there's a huge unmet medical need and we're all very aware. So all any further drugs coming forward would be eligible for all these incentives and everything.
So I'd make two observations, and this is not, I'm going to try and be as factual as possible because I'm not entitled to give an opinion on this panel. But we don't have any research funders up here, but there is a global research funding consortium, and Britain actually has put more of its attention and money into ERDA, the International Rare Disease Research Consortium. So our research money has gone into a global research consortium rather than the European one. That's not completely true, but I think that you asked for hope and you asked for reassurance, and in very rare diseases, I think global collaborations are the way forward because obviously you can recruit more patients and therefore get better statistical power, and you can also, some of the very rare diseases, finding untreated patients is becoming increasingly difficult in Western Europe and the United States. So that's a slightly technical point, but some of the newer drugs actually so if you take, for example, we are going to treat every patient in England with the Atala mutation. Well, we won't be able to do any more trials on new drugs because we're treating. So I think the global collaboration does offer hope. The other observation I would make is that I'm the UK representative on the EU expert group on rare disease and have been for several years, and I would observe that the health collaborations are very flexible in membership. So as for example, Norway always attends. Norway is not a member of the EU, as you know, and a number of the other project groups include countries such as Morocco, Turkey, Armenia, and so forth. So that fact gives hope that there will be a way forward found to maintain collaborations.
That was several. We have time for one more question. There's a question on this side. Yeah.
Sorry, just to come back to the gentleman's point about the if a treatment reduces care costs. In terms of the NHS budget constraints, how flexible are those budgets? And because obviously I'm aware there'll be a pot of money for treatments. If the treatment then reduces the budget, then money comes out of the budget for how much that then has to be spent on care costs for that individual. Does that then increase the budget that can then be spent for treatments? Is there a flow? Is there a connectivity between the two? And if not at the moment, then what is, are there plans to put something in place for that?
Are you asking about social care?
Care across the board, really. Hospital admissions that would go down if certain treatments were administered. Care in terms of maybe equipment that's otherwise funded.
So I'm afraid it's quite a complicated answer, but in respect of the evaluations undertaken by NICE, the comparison is against all the healthcare costs. And then if NICE says yes, that then becomes, as indicated, legally binding on the relevant commissioner, which might be NHS England or might be the local clinical commissioning group. So that's that one. In respect of social care costs, it's more complicated, but the problem of health versus social care budgets is recognised and is being addressed in this pilot you may be aware of in Manchester. So in Manchester, there's a single budget has been created which contains all of the NHS budget and all of the social care budgets of the 10 or 15 district councils in Manchester. So we're all watching that with huge interest, because if Manchester can pull it off and if it's seen to be beneficial, because we recognise that this is a huge problem for patients. This, is it healthcare? Is it social care? If we can make a saving here, does it, that's recognised. The question is whether we can just pull off a single budget where it all works really well, and we all hope it will do, but we need to wait and see.
Yeah, no, thanks. Because it just seems to me that so often the budgets for treatments are just static, and it really does seem that they should be dynamic and more flexible if they come from elsewhere. Thanks.
Well, thank you. That's the end of the session. I really want to thank our panel of speakers who really gave a really good overview, and I think it's a good opportunity perhaps at a later date to do a webinar and to raise the topic again, because there's some more questions obviously that we want to address with the community. So thank you very much to our speakers for coming to present. And we have our coffee break. I'm sure you much need of a coffee now. It's 10 minutes back in here at 3:40. Thank you very much. How do you find that? Are you able to stay or do you have to go?
No, I'm supposed to be doing an interview, another one.
Are you doing that first one?
I did that already, but now I've got another one, I think, with ABPI, yes, a quick one, and then, no, I don't have to rush off, but I can't swim to the dinner tonight.
Okay, no, okay. Yes, she was here earlier. I know. I know. It's taken a few years before we got here. I know. Yeah, it's good. Yeah, we have.
I'm employed in Oxford. I've worked with a number of companies that I'll be talking about, and some of the data that I show you has intellectual property associated with it that I'm an owner of. So I should just tell you that in advance. Okay, hopefully this is introductory for many of you in terms of the dystrophin gene and exon skipping and why this is a powerful method to treat Duchenne patients. The dystrophin gene is encoded on the X chromosome. It's a large gene, but the important features are shown here in terms of the protein that the gene makes at the bottom. And for this protein to really have a good function in muscle, it requires the two end bits to be functioning well. This bit, the N terminus, binds inside muscle cells, and the C terminus binds at the muscle membrane and coalesces a network of proteins. Now, most of the mutations that cause Duchenne result in the transcript, the RNA encoding this protein, becoming out of frame. And by that we mean that the genetic code no longer makes sense. It's unreadable. And as a result, the RNA is extremely unstable, it gets rapidly degraded, and no dystrophin protein is made. So as you can see down here on the bottom right-hand side, this is what a muscle biopsy would look like from a Duchenne patient with very little dystrophin. There's this tiny patch here, a tiny muscle cell that has some dystrophin, whereas a normal biopsy would look like this above, where every single muscle cell is surrounded by a rim of dystrophin. Now, there are many different experimental methods which are now currently available and developing treatments to replace dystrophin, addressing the fundamental cause of this disease. And I'm not going to go through all of these in detail. A number of these target DNA, such as gene editing, which you might have heard about, and the classical type of gene therapy. Others target the RNA, which is what we're talking about here, exon skipping, or the Translana drug, and others target other genes other than dystrophin, but which we think could compensate for the lack of dystrophin, such as utrophin. So in terms of oligonucleotides and exon skipping, why are these good drugs? Why are they probably some of the most promising drugs for Duchenne? Well, these are large molecules. They are a bit like DNA in their structure, but they behave a bit like drugs. So they are much easier to develop than a gene therapy. And this is because we can adapt many of their properties through chemistry. Through change in the chemical structures, we can fine-tune and improve their properties. We can use non-viral systems for their delivery, which is a huge advantage. And the goal of our treatment approach here is really to convert a severe Duchenne type of disease into a much milder and much less severe Becker type of muscular dystrophy. And of course, many Becker patients live for a normal lifespan with a good quality of life with pretty good muscle function. So this is potentially very powerful. And of course, this approach could benefit a very high proportion of Duchenne patients. Perhaps not all, but the vast majority of these patients could ultimately benefit. Now, in terms of exon skipping, this is what the strategy aims to do to correct the underlying problem which I've described to you. So consider a patient here who has a large deletion, a large mutation that eliminates 2 exons from the dystrophin gene. Exons 49 and 50 are missing. The goal here is to develop an oligonucleotide drug that attaches to one of the adjacent exons, in this case exon 51, to modify the way that this RNA is processed, and in particular to modify it by skipping over this exon, exon 51. So this is why we call it exon skipping, and in doing so it has a paradoxical effect because we actually make the mutation larger. The mutation already lacks 2 exons, and now we're going to convert this into a mutation that lacks 3 exons. But the benefit of this is now, whereas this mutation was out of frame, and that's shown schematically by this cartoon here. These exons don't fit together. This new transcript, this new RNA that we've generated is in frame. Exons 48 and 52 fit together nicely, and now there is a genetic code that can be read. Instead of this code, which is unreadable, this code is now legible and can be translated into a dystrophin protein which functions. And because the 2 end bits are intact, which is, as I've told you before, these are the crucial bits of the protein, this is now going to encode a functional dystrophin protein. And so all that this depends on, really, is that this process working efficiently and generating a lot of dystrophin. But the underlying technology and methodology is all optimized to really make this work. Now, all of you will be aware of all of the work that's been done over the last decade or more to try and develop the first two exon skipping drugs. Many of you will have followed the story with BioMarin and their drug, which was ultimately unsuccessful when the US FDA looked at this in December last year. But you will also have followed the story with Sarepta, and their drug eteplirsen was recently approved in September. So this was successful. And I think here it's worth just paying tribute to all of those at Sarepta who've worked to make this successful. And I'd also like to pay tribute to my colleagues here in the UK, in particular Francesco Montoni. I think without this team of people, we would not be here today with the first drug approved. And although, as I say here, I think the clinical benefit from this drug is rather modest, the pioneering pathway that they have developed over the last 10 years will make it much easier for new drugs to come along behind that and be more successful. So this is really an extraordinary achievement, and it will benefit us all in different ways. But as I say, the clinical benefit from this first generation drug is rather modest. The amount of dystrophin made is relatively low. The clinical benefit, albeit important, is modest, and we can and we want to do much better. So what I want to spend a few minutes about is talking about the next generation drugs which are following on behind. Now, the main problem with the first generation drugs is that they do not get delivered efficiently into muscle cells. So here is the inside of a muscle cell. Here are the drugs outside, floating around in the blood and the extracellular fluid, and they do not get taken up into muscle cells efficiently. This is because they are big drugs. They're not like aspirin or paracetamol. These are large molecular weight drugs, and they do not pass through lipid membranes, which is what our cells are surrounded by, readily. This is a major, major barrier. Even if they were to pass through successfully, they need to be in the right compartment of the cell, so they get taken up into things called endosomes. They then need to be released, because ultimately where you want the drug to be active is in the nucleus, where it will interact with the dystrophin RNA and modify its processing in the way that I've described. So ultimately the drug efficacy, the drug potency will depend on how much of the drug is in this compartment within the cell. And the first generation drugs do this very, very badly. Yes, so as I say, this is where we want the drug, in this nuclear compartment, because it's the concentration of drug here and its ability to interact with the RNA in a very efficient way, which will ultimately determine how much dystrophin is generated. So what about the next generation drugs that are on the way? So I see three ways in which these drugs can be improved, and this is reflected in different ways in which people are developing these. First of all, the sequence, the underlying sequence or DNA sequence of the drug, the sequence that it targets within the dystrophin gene, can be improved. I think many of the existing sequences are suboptimal. The sequences can be improved, and I think that's an important thing to remember. The second aspect is the chemistry, the backbone chemistry of the oligonucleotide itself. So I've shown this here by the little arms that link the nucleotides. New chemistry can come along and dramatically transform the properties of the oligonucleotide. And I'll give you a couple of examples here. Aurelie will talk about, I'm sure she'll talk about the tricyclo-DNA chemistry, a completely new chemistry with some very powerful properties. I want to talk to you a little bit about stereochemistry, but there are other chemistries. Chemists are busy inventing new oligonucleotides, and many of these will be coming along in the next few years and could have very, very favorable properties for exon skipping. The third component is how these are delivered into the cells that we want to target, namely muscle, heart, and also the nervous system. I will talk to you briefly about peptide delivery mechanisms, but these are not the only ones. There are new lipid or fatty acid delivery technologies, and of course the whole field of nanotechnology developing in the background is poised to have an impact on delivery. So these are the three components. Any one of these could make an improvement, but I think the real improvements will probably come when multiple answers to these questions are packaged together in a single new generation drug. So very briefly to tell you about stereochemistry. And this has really been pioneered by a company called Wave that you may have heard of, based in the Boston area in the US. And stereochemistry really refers to the shape of the drug. And the way to think about that is that if these oligonucleotide drugs are, for example, 20 nucleotides in length, the bonds between each one of these could be either left-handed or right-handed. In other words, the shape could vary at each junction between each nucleotide. And as a result, if you don't control which shape you have at each boundary, what you end up having is a random mixture of drugs. So your drug, while you might think it's a single drug, is actually a mixture of drugs. And this is a huge problem, and probably was a problem relevant to the biomarin drug, because the number of potential drugs within the mixture is enormous. Billions and billions of drugs could be in that mixture just by having the random property of it being left-handed or right-handed at each oligonucleotide bond. And the extraordinarily clever thing about the Wave technology is that they are able to control the synthesis of the drug so that at each junction it is either left-handed or right-handed. So for example, in this schematic down here would be right-handed, and then 2 left-handed, then right-handed, 2, for example, and they can control the synthesis of this. And so what it means is it's now possible to find out which combination, which shape works best, and then to synthesize the precisely shaped drug that will work best in patients. And we've never been able to do this before. And the Wave technology is truly revolutionary in that sense. They've already begun to work on Duchenne, and their first program is directed towards exon 51, as you might expect, because it's very important for them to be able to show that their drug can have an improvement over the existing Sarepta drug. And here's some data that they have generated in cells from patients who are amenable to exon 51 skipping. And you can see 3 Wave candidate compounds here, all generating enormous amounts of dystrophin on this Western blot compared with the biomarin drug and the Sarepta drug. And when you quantify that, and of course there are always different ways in which you can quantify this, this is generating a huge amount of dystrophin. So one drug 52%, the other one 49%, the other one 100%. But when you look at the improvement compared with Sarepta, this is 70 times better, and over 100 times better. So whilst this is very early, and whilst this data only comes from cells, I think what it promises is the ability to generate very specifically shaped exon skipping drugs that are optimized for their efficacy and also for their toxicity. And so I think my opinion is this is pretty exciting. But we need to see this carried through into animals and obviously ultimately into patients. But this is just from cells. So that's the main caution I would add at this point. The other next generation chemistry I want to talk about really is the work that we've been doing on peptides. And so this really fundamentally addresses the delivery issue, the challenge of getting these large molecular weight drugs into muscle and into heart. So for this, we have been developing a peptide-based technology, many peptides that could serve this purpose, trying to understand what are the optimal properties of the peptides and to find the best one to test in patients. All of this work has been done with a colleague of mine who's not here today. His name's Mike Gate, who's based in Cambridge in the UK. He's the clever peptide chemist. I'm not the chemist. And the idea is that we take the oligonucleotide drug shown here and attach to the end of it a peptide. So chemically attach the peptide that's optimized for enhanced cell delivery. This is the problem we're overcoming. There are a number of ways of synthesizing this compound, but the chemistry is relatively straightforward. To do it in Mike's lab, we do it on a solid phase where the yield is rather low, but to get it up to clinical grade, we do it in liquid phase in large 250-liter vats where we can make large amounts of the compound, and this has been done for us by our Japanese suppliers. And this type of approach is potentially amenable to many different types of oligonucleotide, but particularly those where the backbone is neutrally charged, where there's no electrical charge on the backbone. That is the most straightforward chemical backbone for us to attach our peptide to. And so what we're really addressing here is this fundamental barrier of how do we get these drugs into the cell, and we can overcome this dramatically. We've already identified to some extent what the receptors are that allow the uptake of these drugs into cells, and have shown that we can get dramatically enhanced trafficking of these drugs into cells, as well as increased concentrations of the drug in the nucleus, which is where we need it. Now, when you do this, we can start to see what the benefits are. So now this is data, a number of bits of data from the standard MDX mouse that lacks dystrophin that most of us use in our labs as a way of developing these drugs. And what we've done is shown below here. We've treated mice either with a dose of about 7 milligrams per kilogram of our drug, or 160 milligrams per kilogram of the PMO drug, which is the equivalent of the Sarepta drug. So we've treated with about 25 times less of our drug, and now we're measuring the amount of dystrophin by Western blot, and this is the amount that we can generate in the leg muscle with our drug, in the diaphragm with our drug, and in the heart with our drug. So this is about 10 to 20 times more with a 25 times less amount of drug, which translates into somewhere between 100 and 1,000 times increased activity over the Sarepta drug. So if one could really achieve this in a patient, it would be dramatic. The question is, can we do that? We can show very nicely that we get a nice dose response with the drug. So the amount of dystrophin is dependent on the dose of the drug we get, and we have a good idea that we probably need a dose somewhere around 3-4 milligrams per kilogram, at least in the mouse. But we know now when we do that, when we deliver the dose, these doses of drug, we begin to see a physiological effect in the muscle. So this is beautiful work that's been done in collaboration with Nick Wells, who works here in London, where he measures physiological assay in the muscle, but it's basically telling us about the ability of the muscle to protect itself against damage. And you can see with the lower doses, we get no protection. But as we increase the dose to 9 and then to 12, we begin to see that the muscle has the ability to protect itself from exercise-induced damage, which is one of the major problems initiating disease progression in the patients. That was a single dose of the drug, by the way. When we repeatedly treat mice, we generate enormous amounts of dystrophin, upwards of 60%. And this is probably far more than patients actually require. In this study that we undertook, we calculated how much dystrophin was needed to achieve the protective effects, and it was about 15%. So this is way in excess of what we need, more than likely, to protect muscles. But when we do this, the muscles now are completely protected. There is no loss of strength, no loss of force as we stimulate the muscles, and therefore if you use the muscles, if you exercise, the muscles will be completely protected, which is what we want to achieve in the boys, because this, we believe, would prevent the disease from progressing any further. It wouldn't necessarily restore normal strength to a weak muscle, but it would prevent the disease from progressing any further, which is the first goal that we have. And transforms the way that muscles look. This is a diseased muscle, the diaphragm, where the muscle cells are separated by fat and connective tissue and lots of inflammatory cells, and this is looking like normal healthy muscle, which is obviously why it can function much better. These drugs are also active in the heart, so the peptide technology allows greater penetration into muscle tissue, but also heart tissue. So we get very good dystrophin recovery within the heart muscle. It's not quite as good as we see in the other muscles, but it's upwards of around 40% when we repeatedly treat the mice. And this then translates in the ability to protect the heart as well. So instead of the heart getting bigger or the output of the heart increasing to compensate for that, we can prevent what's called cardiomyopathy by treating the mice and maintain a normal heart size and a normal heart function, even in the mice. And these experiments are difficult to do in the mice because the mice don't have a very severe heart problem, but through exercising the mice, we can generate results like this, which convince us that we can generate dystrophin in the heart and that will be equally as protective as in the muscle. So the way forward for this technology is that the peptides that we have had for about 4 or 5 years are now the proof of principle, are the first generation peptides. And we've been working away for the last 2 or 3 years to generate improved peptides, those which are about half the length, which have improved peptide sequences, which are likely to be safer so that we can develop these and test these in patients. We now have about 3 or 4 series of new peptides, and we hope to be able to select a candidate for clinical development next year. And that would translate into a clinical trial probably before 2019. I would guess now probably in 2018. So hopefully I've been able to persuade you that oligonucleotide therapies really are enormously exciting and have huge potential. It's not just the Sarepta drug, that's the very first step. It's the next generation drugs from Sarepta and elsewhere that are going to have the real impact, and we hope will be transformative in really making a difference to patients' lives. And the peptide technology in particular, I think, offers a very powerful way of overcoming this fundamental barrier, which is how these drugs get into the target tissues of heart and muscle. So I'll stop there. I want to thank everyone in my research group who's done the work, our many collaborators, and particularly acknowledge Mike Gate and Francesca Montoni, who've been helping us with the Duchenne work and development of these drugs. I want to acknowledge Action Duchenne, who've supported us for close to a decade now in the work that we've done, but I should also acknowledge the many other funding agencies that have helped and supported our work. Thanks very much.
I think that we can go ahead with the second presentation. We'll leave all the questions at the end, if it's okay for you.
Do I need a mic? Can you hear me? Yeah, so this one is working. Okay. Good afternoon, everyone. So I think Matthew gave you a really good introduction to the next generation of oligonucleotides, and I will talk to you about another generation, which are called the tricyclo-DNA. So it's different from the peptide. It's just a different chemistry. So this is a chemistry that has been developed in Bern by Christian Lehmann, who's a chemist. So you have the representation here on the right. It's a conformationally constrained oligonucleotide analog, and it has lots of different properties, including an increased hydrophobicity and an increased stability that makes them a really good candidates for exon skipping approaches and the modulation of messenger RNA. So we've tested them in the MDX mouse model, which is the classical mouse model for Duchenne. So just to remind you, this is a mouse model that has a nonsense mutation in the exon 23, and that induces a stop in the dystrophin protein. So if we can skip this exon, for example, by targeting this specific area, which is a donor splice site, we can then skip the exon 23 and have a dystrophin that is almost fully functional. So I'm going to show you some results in this classical MDX model and in also a more severe model, which is called the double knockout model, which is also lacking utrophin. But the first thing when we injected these compounds was to look at, after several weeks of treatment, where we could find the compounds, in which tissues. And you can see here, so in gray and black are two different dosing regimens. And of course, as all oligonucleotides, they mainly go in liver and kidney. But what was interesting is that we could detect quite a lot of oligonucleotides in the diaphragm and in the heart, which is some of the tissues where you need them the most and where the current chemistry, like the PMO, like the Sarepta compound, do not necessarily go very efficiently. For example, they don't go in the heart at all. And we could also detect some in the central nervous system, in the brain and in the cerebellum. So that was something quite unique, and I will come back to that later. They also have some very specific properties, and for example, they can form what we call nanoparticles. So you can see here, this is an electron microscopy picture, and you can see that they form some small particles, and you can also measure this with an experiment called nanoparticle tracking analysis, and this is actually some experiments that we've done in collaboration with Matthew's group. And these are the properties that are essential to improve the uptake and the endosomal escape that Matthew talked about. So this seems to be quite a nice property of this tricyclo. So we've done a comparison study of these tricyclo DNA to the two other chemistries that were used in clinical trials, which are the 2'-ormethyl led by Biomarin and the PMO led by Sarepta. So the three oligonucleotides target the same sequence in this exon 23 mouse sequence. We did injections, intravenous injections of these molecules with different dosing regimen for 12 weeks of treatment, and then performed some different analysis in terms of biomolecular analysis and also functional analysis. And I'm going to show you two different dosing, a high dose and an equimolar dose. You have here the representation of the exon skipping level, and here in black are the tricyclo results, and you don't need to look at all the different tissues, but across tissues you can see that the black bars are always higher. So we have on average 5 to 6 times higher level of exon skipping that leads to also 5 times higher levels of dystrophin rescue in these tissues. And this is particularly impressive in the cardiac muscle, where we have a really good restoration of dystrophin with these compounds, which is absolutely not the case with the two other chemistry, which do not go into the heart. And what was surprising is that we could also detect some of this exon skipping in the brain. So this is a quantification of the exon skipping detected in the hippocampus, the cortex, and the cerebellum of the treated mice. And you can see that we didn't detect anything with the other chemistry because they cannot cross what we call the blood-brain barrier, so they cannot go into the brain after being injected into the intravenously. And this translates also into dystrophin protein restoration. Here you can see in the cerebellum, and here you can't see very well, but also on immunochemistry, if you look at the section of the brain, we have a restoration of protein. So this restoration of protein translates into an improvement of the phenotype. So we looked at different functional rescue. This is the muscle functional rescue. As Matthew mentioned, we can look at the electrophysiology and really the properties of the muscle, of the isolated muscle in mice. And the dystrophic muscles show a force that is decreased compared to normal mouse. And you can see with the treatment, especially with the tricyclo, you can improve this force, and this is similar graph that as Matthew showed, that you can protect the muscle from exercise, from the damage induced by exercise. And we also saw a nice dose correlation, so the more tricyclo you inject and the more force you gain from the treatment, so this is a nice dose response. We also looked at the respiratory function into these treated animals. This is something that's not really done very often in these mice, but they do show respiratory function impairments like we can observe in patients. So there are lots of parameters that are impaired. We can just look at the respiratory function by doing what we call full body plethysmography. It's just putting the mouse in cages that measure the pressure, and you can then measure lots of parameters. And I'm showing here only 3 of these parameters, and you can see that there's normally a difference between normal mouse and dystrophic mice, and in most cases they are restored with the treatment. They can also be restored with the other chemistry, but less parameters are restored with the PMO or the 2'-ormethyl, and the restoration is higher with the tricyclo. We also look at the cardiac function because we had a really good restoration of dystrophin in the heart, and with the high dose we had a restoration of the shortening fraction and the ejection fraction in these mice. So the tricyclo treatment did improve the cardiac function of these mice. What was interesting is that because we detect some dystrophin in the brain, we looked at the potential cognitive improvement, and we're working on this with a collaborator in France who's been specialized in the function of the dystrophin in the brain. And dystrophin is expressed in the brain at the level of the inhibitory synapses, and it plays a role in the synaptic plasticity in the brain. And what is interesting is that in the MDX mice, they do show some defects which are correlated with the stress. So they have an enhanced response to the stress. So the stress response is really different from a normal mouse. And we can quantify this as a freezing response. So if we stress the mouse, they spend a lot of time frozen in the cage, so they stop moving. And this is not a motor function, this is not a muscle issue, because if you don't stress them, there's absolutely no problem. So the wild type spends only 20% of the time not moving in the cage after a stress, whereas the dystrophic mice spends 80% of the time frozen. And when we did this test with the different treated animals, we could have a restoration of the normal animal's behavior with the tcDNA treatment and not with the other chemistry. So that was really correlating with the level of dystrophin that we could restore in the brain. We also did an evaluation of these compounds in the double knockout mouse, as I mentioned. So this is a useful mouse because it is much more severely affected. It has a lifespan that is much reduced of about 10 weeks. So we looked at the skipping efficacy in these, when does not appear? You don't see them. Okay, so you're supposed to have graphs here of skipping efficiency and functional rescue. So we did obtain a really good efficacy of the tricyclo in these mice. And what was impressive is that it restores the phenotype. And you can see it very obviously in these mice because they develop a very strong kyphosis on the back. And as soon as you treat them, they look like normal mice and they behave in the cage as normal mice. So the tcDNA treatment also restored a very severe phenotype. So the first conclusion about this study was that tricyclo shows a superior efficiency compared to the other naked chemistry. And it improved the respiratory function and the cardiac function as well as going into the brain. So it shows a good potential for the systemic treatment of Duchenne. But now what is important for a new chemistry like this is to be safe. I mean, this type of chemistry has never been used in men. So we did an evaluation of the toxicological profile on the high dose for 12 weeks. I'm going to show you quickly some of these tox profiles. So the first thing we looked at was the short term. So after an injection of this high dose in mice, so I don't see any graphs. Okay. So believe me. So we looked at all the cytokines in the blood after 1 hour after an injection, and there's no changes. So all the inflammatory cytokines are not elevated or decreased. So the profile is not changed. And we then looked at the long-term chemistry. So most of the toxicology is associated with the oligonucleotides is linked to the accumulation in tissues, and in particular in liver and kidney. So we looked at the oh my God, you don't have any graphs. Okay. So it's going to be short. So long-term chemistry. So we looked at different enzymes in the blood. So we look at the transaminases for the liver function. We looked at the creatinine and albumin level for the renal function. And the little star that you can see here is only a very slight increase in the creatinine levels in the kidney, which is very moderate and which has also been demonstrated at higher levels for 2'-O-methyl. The kidney and liver histopathology looked really nice, so we could only demonstrate a very small changes which are considered really safe for this type of dosing. So when you inject 12 weeks with this type of dosing, you do accumulate a lot of oligos in the kidney. So showing such small changes was actually quite a good news. We looked at the urinary profile as well, so we collected urine and looked at different biomarkers in the urine, and we didn't see any changes in the total protein or the creatinine, so imagine the graphs. And And then we finally looked at some very specific biomarkers. So you can look at things that can change dramatically in the renal function. And actually you're looking for these biomarkers because you want to see something that is going to be able to tell you at what dose you start to be toxic. This is something that you're looking for. So we looked at some biomarkers like KIM1, which is a very sensitive biomarkers of the renal toxicology, and it can change from it can go up to 200-fold when a molecule is toxic. So I guess you don't have the graph either. But with the KIM1, we obtain a very small fold changes, about 10-fold after 12 weeks of treatment. So that's the early sign of an accumulation of oligo, but it still stays very, very low compared to what can be achieved at 200-fold. So overall, this is a very promising toxicological profile for this tricyclo. So they have always been very well tolerated in all treated animals. We didn't detect any significant changes in the serum biochemistry or in the urinary biomarkers, only the very specific ones where we start to see a very small change after a really high dose injection. So we're currently developing and validating the human candidate. So we're working with a small company who's producing the tricyclo. It's called Syntena. It's based in Switzerland, in Bern. So it's producing the candidates that we're working on, and we're validating them in cells from patient and also in mouse model that have a deletion of the exon 52, so we can really confirm the efficacy of the clinical candidates. And we're evaluating the specific toxicology profile of these sequence. And so we're also working on more fundamental aspect, trying to understand more the binding protein, the binding properties of these molecules, trying to explain how they can cross the blood-brain barrier and how they can be efficient in going into the heart, for example. So I'd just like to thank people from the lab. So we're working in an interim unit directed by Luis Garcia, so some of you might know him. And this is the group who's leading all this study, and all our collaborators. And then you should have all the logos as well of the supporting agency. But so this is all our international collaborators. And thank you for your attention. And sorry about the slides. So I don't know if Matthew wants to come back and we can discuss all together. Or...
Hi, I thank you for the very encouraging talks that you both gave. I have a question to Matthew Woods, if I may. Sorry, I'm here. So you presented to us two different approaches, the wave one and the one that you're developing. Do you see them as being competing, or is there a chance of combining both?
Yes, I think as I tried to explain, I think there are three ways in which the next generation of drugs can be improved, either by improving the sequences, and I don't think enough attention has been devoted to that, developing or improving the chemistry, and then addressing the delivery. And all of these could be combined. So the two examples I gave, one of which was an improvement to chemistry, and then the peptides, which is really addressing delivery, in principle could be combined. And ultimately, I think you'd want an optimized sequence, the perfect chemistry with a method for delivery that overcomes the challenge. And then, and hopefully in less than 100 years' time, we'll have the perfect drug. Hopefully it's not that far away, but it's combining all those three components that will give us the very best drug. So they are synergistic. They can be combined.
Thank you. Can I have a second question to Aurelie, or is it?
Yes, of course.
The crossing of the barrier barrier in the brain. Do you think that may be a problem if we get to regulatory states?
No, I don't think so, because it is at very low levels. So and it's something that we see with quite a high dose of treatment, and then probably in patient we wouldn't go with such a high dose. So I think the passage would be really, really low, and we don't see it as an issue. We see it really as a benefit. So because it could treat some of the low impairment. So.
Yep, sorry. This is a question for both of you. So far we're talking about exoskipping being a treatment. With this impressive analysis you made, can we talk about cure?
Sorry, I didn't get it.
Can it be a cure rather than just a treatment? Second generation.
Well, I don't think so. I think we can even with the second generation, we can stop the progression. But in order to cure, I mean, we're always going to be converting with exon skipping strategy. It's always going to be converting Duchenne into Becker. So even if the protein is really efficient, it's always going to be a Becker muscular dystrophy. What do you think?
Duchenne to a severe sorry, thank you. So yeah, but with the same strategy, how can you be sure that you are not converting a mild Duchenne to a severe Becker? So actually make things worse.
Yeah, I mean, well, to answer that question first, I think it's possible to get some indication as to what the dystrophin, the new dystrophin would look like by looking at the corresponding Becker phenotype. Many of the Becker patients already exist, so I think one would know what the quality of the dystrophin would be like and what the Becker phenotype would be like. So, you know, in theory you're right, but in practice I think the chances are very, very low. But I would be a tiny bit more optimistic than Aurelie. I think, you know, talking of a cure is not impossible, because I think combining many of these improvements and then getting to the point where one could treat patients at diagnosis, which is the goal, really, not to treat patients when they're 6 or 7 years old or older, at birth or at 1 or 2 years old. And I think then if you can slow or dramatically slow progression of the disease, you're doing so in relatively healthy muscles, very healthy muscles, rather than working with muscles that have already begun to suffer from 5 or more years of disease. So I think it's that combination as well, of being confident enough that the drugs could be administered very early on in life, as soon as patients are diagnosed, because once patients know they could be treated early in life, they're going to be all the genetic tests for the prenatal diagnosis that will come about. I mean, these should be happening as well in parallel, so we'd be able to identify patients earlier and treat them earlier. So I think the combination will really result in dramatic benefits.
Another question?
Hi, Matthew. You talk about various different ways of combining the 3 potential therapies. Does that mean that each and every new compound or new therapy drug you come up with is going to need a whole new clinical trial itself?
No, not necessarily. But I think, you know, clearly if you develop a new drug that is a new combination between a new sequence and a new chemistry and a new delivery method, it will require clinical trials. I mean, there's no getting away from that. But I think if you, we've both described, if you develop a tricyclo, which is an improved chemistry, or if you develop a peptide with an existing chemistry, which is the PMO, then, you know, you'll have to do some very good clinical trials at the outset, but subsequent drugs, you know, should be able to be developed more rapidly. But, you know, once you combine more of these, if you bring a new chemistry, then of course it's going to need a very careful clinical study, because we want to be sure that there's going to be benefit and not harm to the patient. So that's, yeah, no, I think that's where the follow-on drugs, I think, you know, our expectation is that if we can provide data to the regulatory authorities that the first drug we test has benefit and is safe, we should be able to get more rapid paths to developing the follow-on drugs. But it's going to always require at least one drug that has a good set of data that convinces us all that it's safe, and then we should have a very strong argument to say we want to develop the next one even more quickly, and the next one, and do them in parallel as quick as we can. But there's no getting away from a very, detailed clinical study at the outset.
Can I just ask a quick question, which is, I know that is a tricky one, but can you give us an idea about timelines in your, I know that it's very difficult to predict, but just to have how many steps we are.
So with the Centella, thinking with the tricyclo, they hope to go into clinical trials maybe in 2 years, the time to have the filing, and then all the finalization of the tox study in rats and monkeys. So these studies take a long time. So yeah, probably 2018.
Yeah, I would say 2018-19. I think the reason why one can be more optimistic is it's because of all the work that's been done in the last 10 years. So we know roughly where the goalposts are and how we get authority to do clinical studies and what the design of a study needs to look like to be persuasive. So I think it's easier. It's still a big challenge, but I think we can be quicker than we used to be.
Thank you very much. We need to move on to the next section. So in the next session, that is also the last for today, before then the gala dinner tonight, is really an important, I think, issues which I think is very important to discuss, and is about how do you have the best transition to adulthood. I think that is something that all of us will benefit from. Will be really a talk about the lesson that has been learned from Takin Charge project. I don't have one presenter, but it's more, I think, a collaboration in the term of the presentation. So thank you very much. All right. The USB drive over there is working. Are we going to get everyone ridiculous? Do you have one as well? Yes. Oh, do you? Okay. Sorry, I couldn't email it. Do you want to move anything? Actually, right there. Just here for a while until it's your turn. Tidy. Sorry, final. Jack and Aaron, are you coming up to talk? In fact, I'll save it at the end. It's fine. Yeah. That's all. Where's the time? You all right? How are you? I'm hot. Yeah. It's cold in the other room. Well, we turned up the heat. Removable this. Is it? Hmm? You okay? I'll get you a. As well. Oh yeah. Is there a roving light for a minute? In a minute. Hold on a minute. Sorry, I just want to. Yeah, I need to put it on the slide.
Mitten, what's it called? What's your key called? You've got it there, yeah?
Okay. Can't see that, my glasses on. Okay, thanks everyone. You've lasted this long. I'm sure it's been a really long day, although I'm sure you've all learned a lot and had opportunity to hear from other families and amazing experts. We're going to round off today really just to celebrate the project that we were doing for the last 5 years, which was working with some of these guys here on the stage and also a load of other people who are getting ready for transition, which we mean between the ages of 14 and 19, so getting people to think about their futures. So you're not going to hear just from me and Celine, although we were kind of running the project as managers. You're going to hear from our fantastic steering committee who helped us to organise and develop the project, and you're going to hear from the boys themselves who took part, and you're going to hear from the parents. Okay, so so today then we'll be hearing from me and Celine very quickly about an overview of the project. Vicky and Wayne, over there, are going to be talking about being parents on the project. Jack and Aaron and Saul will be talking about being young people. Judith will talk about some of the parent sessions and what it's been like being a parent of a boy going through transition. Mithan, who's been part of our steering committee, will talk, and then we'll be hearing from DMD Pathfinders, who are going to talk about plans for the future. So, do you want to do this bit?
Okay, yeah. We're very rehearsed. So just for those of you who didn't know what we were doing for the last number of years, or don't know, Takin Charge won a lottery bid, and the project was for young people 14 to 19 years of age with Duchenne, and it was thinking about transition, and it was delivered through a accredited program in workshops and an e-portfolio, which we're going to let you see in a little moment. And it was all about preparing for adulthood. When you think about being an adult, what kind of life do you hope you're going to have? It also involves sessions we call it letting go for parents, and also some sessions about what about us for siblings, because we knew that siblings and issues for siblings are something that has to be looked at. And we had, of course, the great foresight right at the beginning to bring in the advisory team of the steering committee, who were Mark, John, Jonathan, Stuart and Mitten. And we just sadly need to say that Jonathan and Stuart are no longer with us, but their support on the project was really, really influential.
Okay, so some of you have come to education sessions today, and really the launch of this programme coincided with some new legislation that was happening. In 2011, there was a green paper called Support and Aspirations, and in 2014, this became the Children and Families Act. And really the focus of this legislation was all about getting a life as a young disabled person. So it was perfect timing for this project, and perfect timing to get our heads around what it was that we need to be thinking about, and how we could use the legislation to our favour, really. And the new legislation looks at planning and people's lives in 4 areas. It looks at employment or education as the first one. It looks at how you're going to be independent and what your living arrangements are going to be when you grow up. It's going to be about your community inclusion, what friends you've got, and it's about getting in control of your health. So when you're planning for adulthood and you have your education, health and care meetings, these are the 4 areas that you'll be talking about in your meetings at school or college, and you actually have a legal right to talk about these things. So in the past, people might have said, oh well, you don't want to be getting a job, or oh no, you don't want to be going and getting a flat. But now there is a legal right to discuss these things, and that's really, really important. And we've managed to talk about these things with our families and our young people and get them to think about what it is they want to be seeing in their lives. Okay, so the focus of the project then has been about encouraging independence, developing skills that could lead to maybe college. Some people want to go to university. Some people want to look at supported employment or supported internships. So there's a whole range of options. We're not saying everyone has to go to college, everyone has to go to university. It's really starting with a young person and finding out what it is they want to do. We've looked at understanding my condition. Some of you might have seen our animation that we did with some fantastic animators, and the boys helped to make Understanding Duchenne Muscular Dystrophy, so Google that if you haven't seen it. So through that we developed our IT skills and developed the ability to animate. We've made friends, we've looked at sexuality. Do you want to talk more about that?
Well, some of you were at the session just before. What we did was we worked very closely with other organisations who were thinking about the area of sexuality and disability. And certainly through the steering committee, we were able to influence a lot of the thinking around sexuality and disability and guidance that has been produced. But in the sessions with some of the young people, we also had very interesting sessions where, you know, we answered the question, are you going to tell us how to do it? Are you going to tell us how to do it if you're in a wheelchair? Because that's a question that young people ask, and it was part of our programme.
Okay, and through, so the project really was about looking at various challenges, and we did this through a kind of structure of an e-portfolio, and boys kind of followed certain challenges. In some places across the country we followed this to the T. In other places we took the spirit of it and looked at different areas. So we looked at, I can't read this without my glasses on.
Okay, we looked at Future Me, understanding your condition, how using assistive technology can support you in adulthood, the importance of health, mental health and well-being, and having good leisure and good life, but all about planning for leaving school and the preparation through the transition years in school to have the life that you wanted as an adult. And it was presented as a kind of a challenge. So some of you know about the Duke of Edinburgh Award, and so, you know, things that people might not have thought to do, they were being challenged to do. And you're going to hear from some of the young people here today about some of the challenges that they took on and things that they've achieved that they didn't think they might do at the beginning of this programme.
Okay, and here's some of the things that we did do on the project. So here's Sean. I don't know if Sue's here. I can't. Right. At Hereward, where we were doing some animating and some IT skills, we went to Parliament, invited all our MPs and asked them to come for afternoon tea and told them about what our aspirations are for the future, so that when they were passing this new legislation, they were aware that there were young people who had medical needs who also wanted to be included in it. And so as a result of that, I think, as well as other lobbying, there was a section of the new legislation which is about medical needs as well as specific educational difficulties. And here we are in the Olympic Park at the bottom there, just having a Taste of Disability Sports Day. Yeah, shall we hear from Vicky and Wayne now then? Yeah. So work that we did with parents, particularly with parents, was looking at things like person-centred planning. We looked at how to do person-centred planning because this is very much about what the new legislation is expecting us to do. It's expecting us to think about our futures and is expecting us to be able to identify what it is that we want to do in the future through a system called person-centred planning. And we did a lot of preparation work with this with families. Vicky and Wayne, who have Aaron and Jack, are going to now talk to us about being parents on the programme.
You got copy. You go. In other words, I'm going to do all the talking, and you're just going to stand there looking pretty. Okay then. What Takin Charge did for us was basically enable us to find out about the new legislation and what it meant to us and what it says, helping us to make the decisions for our boys. It actually helped us to link up with people in the same situation, so all the guys behind us and a lot of the families, some of them who are not here today, to actually discuss with them, put our ideas forward, and also use them as a shoulder to lean on, and also turn to them for advice as well. Also to be positive about the future, what we did and what we wanted to find out, and what we wanted for our sons, and what our sons actually wanted to do with their lives, and basically just to say yes to your boys, because they are our future. And
with the help of other families and ourselves, our boys can actually go out there, plan what they want to do with their lives, and basically realise that they have got a future, and basically give them the tools to actually look at what they want to do, how they're going to do it, and then basically just take charge of their lives and ask them when they're 25, what do you want to do? What kind of life do you want? Aaron, he turned around, and Jack says, don't want to learn to drive. People turn around and says, you're crazy. We looked at him and we says, no, you're not. You're going to go out there, you're going to learn to drive with the help of ourselves and with the help of Motability. Aaron and Jack have now got cars while Jack's waiting for his car. Aaron's just passed his driving test and he's gone out there in the big wild world. He's showing people just because he's got Duchenne and a muscle weakness, he can do things. Also, Aaron wants to become self-employed and we're helping him along that pathway as well. And also develop friendships, not just for Aaron and Jack, ourselves as well. We've made a lot of good friends along the way, and without Takin Charge, we wouldn't have met a lot of the people we do know in this room. So I'd like to say thank you to Celine, Janet, and the rest of the Takin Charge, especially the young guys, what you see behind us, and the young adults, because without them, it wouldn't have been a success.
It's important to have access to older guys with DMD, to give the advice and guidance that the younger men will need for their future, really, because the older ones are doing it already and showing them that, yes, there is life out there and they can have one. And it's important to have support networks during transition and knowing just to talk to people in the same position as yourselves. That's really important because we're all trying to fight the fight for our boys, really. Do you want to come over? Aaron, do you want to come first? Aaron, come first. Okay.
So what bit am I reading? That bit at the top. Think about the future you want and be prepared to talk to people about it.
Aaron, do you want to?
So when I joined Takin Charge, I said in my first session that I wanted to learn to drive. With the help of my parents and support from Motability, I can do it now. I wanted to be more independent and not to have anyone to rely on. Anyway.
Okay.
I wanted to be free to go where I wanted to go, when I want. It gives me a sense of achievement in doing something I thought I couldn't.
Okay. All right. Just this bit. Yeah. Just speak a little bit louder.
I also wanted to have a job in fashion. Working with the British Association of Supported Employment and Birmingham School of Fashion, I saw that it's possible, and I am on track to achieve my dream.
I don't know why the photographs aren't coming up. Do you want to come?
Oh, if you click it.
What do I need to do?
Click them up and measure them.
Click the both, sorry. No. Sorry, there were lovely photographs of Jack
and in the School of, or Aaron in the School of Fashion. Okay, Jack, do you want to, and you were at, do you want to come and talk about Barrett Homes? No. Do you want to come and have a look?
I don't know what to
Just talk about the assisted, what you did in Barrett Homes.
I've done a work placement at Barrett Homes and I learnt new skills. I learnt how to do CAD, like designing houses, which was interesting. Yeah. After working there, doing 2 weeks of work experience, they offered me a job. A top tip is prepare for any transition meetings and be sure to say what you want your life to look like when you are an adult, what you want to be doing for a job, where you see yourself living, what relationships, friendships you hope to have, how independent you hope to be, how you want, get it all on your EHC plan, how you want support for getting a job, how's living independently, how talking to clinicians and knowing how to keep yourself as healthy as possible as adults, having a social life relationship and being part of your local community.
I don't know what's happened, but the photographs haven't come up, but we had wonderful photographs of Aaron and Jack taking on the challenge that they set themselves last year to do a sponsored wheelchair event. And in this occasion, it was from Bristol to Birmingham, and that was a major achievement, and they deserve real credit. And they're an example of young people deciding to take on things that no one thought they could do, and they have achieved that. So well done. So.
I don't know if the photos come out or not. Do
you want to come?
Just go there and look at the big screen, or?
Can you see that, Saul? Yeah. Well, why don't you come and read it from here then? Yeah. Yeah.
Over the past 2 years, I've taken part in lots of different workshops, such as making protest beats for the lobby of Parliament, game design workshops, how to create games, and thinking about what I want to do for a career in the future and the skills I would need. Me and my friend Alex took part in an inclusion meeting in Luxembourg, which we applied for. This was funded by the Department for Education. I stood for Head Boy. This involved running an election campaign with which my sister helped me with. And I made a speech. I won. When I was Head Boy, I made changes to the school canteen and set up a link with a refugee camp in Palestine. Me and my dad set up a petition to get NHS England to fund a genetic treatment for Duchenne. I presented this to David Cameron in January 2015. This was finally agreed in May 2016 so that boys who can benefit from Translana can now get it on the NHS. In Taking Charge, I thought about my future and did work experience at South Bank University. Here I am in the games culture department with some second years who I worked with on a project. When I'm older, I would like to go to university to study game design so that I can be a game designer and set up my own game design company.
It doesn't. Okay.
I got 9 A to C GCSEs. I'm now at Leighton Sixth Form College studying software engineering. I hope to go to university to do a course in game design. Not bad for a boy who struggled to learn and read and could never do maths.
Right, Judith, which one was yours? Open it up wide. Don't know. Is that it? This one here. Hello, everybody. I'm Judith. I'm a mum of Sam, who's just behind me. He's 19, and we have survived the transition process, I think. But I put this slide up because it's coping with change. And to me, Duchenne muscular dystrophy is all about change. And, you know, every step along the way, there's change. And I look back in the last 11 years since my son was diagnosed, and I kind of look back and think that all the changes that we've gone through, we've actually survived those changes. So it gives me, if you like, the strength to carry on and deal with all the changes that are coming. And when we talk about change, you know, from a parent's perspective, it's very hard, and I can't lie, I found it very hard, that transition process of letting go as a parent of a child with DMD. But I actually had to force myself to think, well, okay, my child has DMD, my young person, who's a young person now, has DMD, but why should he live a life differently than a non-disabled person? Why should he not have, you know, the ability, the chance to live independently like any other young person? Why should he not have a chance to work, to drive a car if he wanted to have a girlfriend, to whatever he wants? Why should he not achieve his dreams? So, oh, how do I get the next slide? Sorry. Right, I don't, you tell me what to change it. That's it. So I thought to myself, okay, so what's the difference between a disabled young person and a non-disabled young person in terms of letting go? It's very hard for a parent to let go, whatever your child, whether your child is disabled or non-disabled, it's a very emotional thing. But I know this sounds really corny, but if you love somebody, you have to let them go. You have to let them make their own mistakes, learn from their mistakes, and build their life. And this is what I'm doing with my son. You know, there's no manual in bringing up a child, let alone a child with DMD. So, you know, I just wanted to say today that, you know, each case, I found that, you know, each case is unique. You have to do what's right for your young person. Your young person has to do what he wants to do. You can't compare yourself with anyone else who has Duchenne. Everybody is different. And, you know, each case is unique. And you just have to be there as a parent just to support your child, whatever path they want to go down. So Sam, he's, well, a work in progress.
He's at university. He's in his second year at university studying journalism. He's living away from home independently in halls of residence. And for him, it's a big step, and I'm really proud of what he's achieved. And I've noticed that this year, his second year, he doesn't text me as much. He doesn't involve me in much with as much as he did in the first year. So it means that things are happening. He is taking ownership of his life, and that's all I want as a parent. As you know, I think any parent wants that for their child. Taking charge. You know, all this has happened with a lot of help from Celine, Janet, and Taking Charge. All the other parents I've met, all the other boys, young people with Duchenne that I've met, and it's taught me that, you know, along the journey of Duchenne, with the journey of Duchenne and being a parent, you have to have patience. Things do happen, but they happen slowly. But if you have the determination, things will come about, and positive things can happen. And when I look back on the last 11 years, I've had very pleasant surprises on this journey. And, you know, Taking Charge has helped with sharing advice. I've learned so much. I've learned so much today. It's just overwhelming. And every day I learn something, because advice is shared and support is offered. And that is really important as parents, you know, because we all feel the same. And belonging to a community, it's like belonging to a family. And I know that I will always belong to this family, and I will always have support as a parent. And so will my son. I've nearly finished, but you just need to, and I've learned as a parent that you need to compromise and adapt. Nothing's perfect, but you try and make it as perfect as you possibly can. But, you know, you just learn to compromise as well. That's important. And, you know, being with Taking Charge, you know, I know it's important to never give up, because there is support out there. And, you know, I've found that support, and so has my son. And, you know, whatever the future holds for Sam, I know that he has, you know, he has the confidence, he has the self-esteem to know that he has the right to live an independent life, just as anybody else. Thank you. Thank you.
Yeah, so hi there, I'm Sam. Yeah, I'm 19. Yeah, I haven't got any presentation to give or anything. I'm just going to chat about yeah, my role, what I've gained from Taking Charge, and what sort of I hope to do with in the future. Yeah, basically, Taking Charge, the project, yeah, has been about trying to improve quality of life as one of the main goals in terms of socially, sort of employment, education, and that sort of thing. And yeah, that everyone has the right to achieve those sorts of things. So yeah, now that that's finished, I think the sort of aim is for DMD Pathfinders to sort of take on that as a user-led organisation. So providing advice from the people with the best expertise. I'm hoping to be part of that in the future as the organisation progresses. Yeah, at the moment, obviously, one of the key aims is to enable people to have goals and do what they can to achieve those goals. So yeah, I'm at the university, currently studying journalism, hoping to go into that industry. So yeah, I'm focusing on that at the moment, taking one step at a time. But yeah, I feel it will be important at some point when I have some time to give advice to maybe younger people with DMD, 3, 4 years younger perhaps, who maybe want to have similar experiences going through a transition stage that I went through. I hope successfully. I feel like now it's becoming successful, and yeah, I think DMD Pathfinders can have a real good role in giving support to others at different stages of their lives. I know when I was 3 years ago, I think I came to this conference in in 2014, and listening to some of the older people with DMD like Mark and John, it was incredibly useful. Gone through similar, through they've had a lot of life experience, and I found it incredibly useful to speak to those older people and to help me feel motivated to achieve things. So I think it'll be really good for the organisation to sort of follow its mentor. I think there's going to sort of hopefully start a mentoring scheme where to give advice and share things. I think that's going to be really useful. For me personally, I found my first year very much a challenge at university, getting used to independent living, for less being reliant on parents to sort of being reliant on carers. But I kept motivation because I knew that there's people with my condition that have done it before. So I think a good way forward will now be for adults with Duchenne to start giving advice and things like that. Thank you very much.
Just while Mitten's coming forward, I mean, when we started Takin Charge, we were offering support for transition, and it was in the process of doing the project that we realised the leadership that was coming from adults that was influencing the information and that we were sharing with young people and families. And we suddenly realised actually what we needed to be doing was supporting the adults to facilitate the workshops, to give the information to the families and young people, because it was so much better coming from them. And so you'll appreciate that what we've then been doing is developing that leadership, so that you've heard Sam say he's in a position now where he wants to cascade that down to the next group coming through. And we had a wonderful leadership camp in September, which was about handing over to a really brilliant cohort of young people who, having gained so much themselves from this project, recognise the value of it, want to give something back, and have developed skills through the project to be able to do that. And one of those is Mitten. Can you see it, Mitten? Just give me a minute here.
Yeah, I'm going to talk about my journey to employment that I gained from the Takin Charge project. Yeah, I started off by volunteering for various different charities, as well as Action Duchenne's Takin Charge project. I was an Acorns ambassador, West Midlands Regional Trailblazer ambassador, and a trustee for the Pathfinders. Yeah, the skills I developed my confident skills, public speaking skills, and the ability to mentor young disabled people. Yeah. I registered as self-employed as a disability consultant by I started working for Action Duchenne. I've also been doing game testing for the London South Bank University, and recently been offered a job as an expert by experience from Together for Short Lives. And I also have a small photography and videography business on the side for MS Studios. Yeah, and I sold this picture to my school, the school that I used to go to, Wilson Stewart.
Okay, that's your photograph of the bull ring, yeah, in Birmingham. Okay.
Thank you for listening. Yeah.
Thank you, Nathalie. you ready? I'm sorry we didn't get the photographs in a number of the presentations, but nonetheless, we hope that we have managed to convey what we consider to be real achievement through Taking Charge. But we know as well that it's only the beginning. And, you know, Janet and I have worked hard on this, but we're also really delighted to be handing over to DMD Pathfinders, who at the beginning of this project didn't exist. No one thought that they could or even should exist. And in the process of this project, I think everyone has come to realize that what is needed in this organization, that is the community Duchenne, is adults speaking for themselves. And I'm going to hand over now to John and Mark to talk about how they're going to take things forward.
Thank you. Yeah, I think it's worth just saying, so the steering committee that was formed for Taking Charge was basically where Pathfinders came from, and I think without Taking Charge there wouldn't have been Pathfinders. I think what I've really felt is it's just completely, it's been transformative, Taking Charge. So I think looking back to my childhood and remembering, you know, friends with DMD and just the complete lack of expectation that there was on anyone. And I think, you know, it's really quite rare to find someone that had gone out and done something with their lives and kind of realised their personal goals. And I think now, like looking at all the people involved in Taking Charge, you know, they've all done amazingly. And I think, you know, it's no longer the exception. And I think it just goes to show that what can be done if people are given the right level of support and encouragement. I think, you know, Pathfinders are very keen to take this work forward, and Mark will say a bit more about that as well. But what I wanted to just kind of emphasize is Taking Charge was, I guess, kind of proof of concept. It was the human trial, and now we need to kind of roll out this idea to the wider population. So I think, you know, it was funded by a lottery grant, and I think, you know, to suggest that we can just pick up where Taking Charge is finished and carry it on as it is with minimal effort would be to give the wrong impression. You know, it's going to be a challenge, but we've seen just what an amazing difference it makes to people's lives. So we're really committed to kind of taking it forward. But I think I just wanted to say that because I want to say that we need all the support that we can get to make this happen. DMD Pathfinders, it's a fledgling organisation, so, you know, we need time, we need support, and we need, most importantly, money. And we're, you know, we've got ideas for how we can, like, address all those challenges and move things forward. And I think, you know, the difference that we can make if we can make this happen universally for everyone in Duchenne, that could just be absolutely life-changing for so many people. And I think what's also so amazing about Taking Charge is, you know, it can be applied to everyone. It doesn't, you don't have to be super academic and wanting to go to university. I mean, that's one option, one pathway, but, you know, you might want to start your own business. You might want to get another job. You know, there's all sorts of things that you could do. And I think it's about having personalized support for everyone. And it's about that being driven by the, you know, by the experts, by the people who've got the lived experience of Duchenne. And I think, you know, we're in a unique position to be able to take that forward. And I look forward to doing that. So I'm going to hand over to Mark.
Thanks very much, John. I think Taking Charge has been incredible, and it has taken us all on a journey, and I think the end result of DMD Pathfinders is a huge achievement that has come out of it, and it's only thanks to those of us doing behind the support scenes that really it came into fruition. And really, I think we really view it as it's our ambition is really changing the outlook for the future of what you can achieve with Duchenne despite its limitations. And I think we're really wanting to push our organisation forward, and really we're looking for other guys with Duchenne to get behind us, and those that would like to, we would like people with Duchenne to commit to the organisation. Whether that's just doing small things, like doing tweets on social media, or even bigger things like becoming a trustee, which is something that we're really needing to get committed people on board as volunteers and also as trustees. And really, I would hope to see the organisation grow so that it can actually influence and make positive change for the future. And I think I'm really pleased that DMD Pathfinders come about, and it's really, I see it as a natural progression on from Takin Charge, which did so much for young people and their families, and I'm really pleased that to have been part in that. So I'd like to thank everyone that's involved, and you'll be hearing more of DMD Pathfinders in the future. Thank you.
We're hoping to, that's it now really, but we just wanted to say that we'll be working on a kind of leaflet on top tips for transition that Celine and I will create in liaison with Pathfinders, so that we can people can learn from our experiences of doing this project, and from parents and young people and siblings. And also we did a sort of a formal evaluation of some parents over the last year, and that's going to be published in the British Journal of Special Education next year. So we'll advertise that through Action Duchenne as well, so that you can use that to show people at college or at school. And it just shows how it's really interviews with parents about their feelings about transition and what's helpful and what isn't. So thank you very much.
Before everybody goes off, I think we are just waiting for one more person. Is Diana here yet? No? Oh, somebody's running to see if she's there. No. Okay, doesn't look like Diana's here, so I'm going to have to hand over to my second in command, Ravi Mehta.
Yeah, on behalf of everyone who was a part of Takin Charge, everyone at DMD Pathfinders, all the families, we want to say a special thank you to Celine and Janet. You know, it was the first time someone asked all of us, what do you want to do as adults? Where do you want to go in your life? And I think you've empowered a lot of us to where we are today. So yeah, big thank you, and we've got a little gift for you as well.
Come on, Janet.
Just hearing you say that, Ravi, it was the first time anyone had asked you what you wanted to do when you were an adult. That just says it all, really.
No.
Sorry I'm late, everyone. I was just running from another session, and I wish I was in this session because Janet is a fantastic member of the community. She co-founded Action Duchenne. She's an expert, and I think we have a lot to be grateful for. And obviously you're still going to be a fantastic member of the community in the future. And we just really want to thank you on behalf of the Action Duchenne team for all that you've done for us. And equally, we would really like to thank Celine for everything that you've done with the Taking Charge program. Invaluable member of the community. The adults and young people love you, and I think they will really miss you. So please do keep in touch, and I know that you'll be working with us in the future. Thank you so much. And yes, just a quick word. We've got our fantastic gala dinner, and I'm delighted to say that Harry Hill, our patron, will be joining us and providing some entertainment at 7 p.m. So you must come to our dinner. If you haven't booked your place, do come find a member of staff. Harry Hill will be there for an hour, and he's delighted. He really wants to meet you all. So it's fantastic. It's going to be our best dinner yet. So don't miss out, and a fantastic end to a great day. Thank you to all our speakers for today. It's been really amazing, fantastic sessions. And a big thank you to you all for coming, and a big thank you to our sponsors. And for those of you who missed the World Muscle Society session today, Perspectives from a Researcher, we are just going to do a very short presentation as well in this room if you want to hear some more updates. So if you do want to stay and listen to the presentation, you are more than welcome.